I'm delighted again to introduce Dr. Angela Dispenzieri, who is the chair for the Endpoint Development Working Group. So I'll turn it over to Dr. Dispenzieri.
All right, so thank you. Okay, so this is on behalf of our group. You know, endpoints have sort of three major roles here, right? We use them in the clinic, one patient at a time. We'll have endpoints that may be unvalidated endpoints that may be used in small trials, but most importantly, or at least for today's discussion, it's really the endpoints that are validated sufficiently to satisfy regulators so that we can get new therapies to our patients. So this is the Endpoint Steering Committee membership. So what we did is, because endpoints are sort of cut across many different things and require many different specialties. It's not me. Require many different specialists and specialties. We sort of broke them up into subgroups. I have to thank our cardiology leadership. Basically, oh, is he on? Yeah, Justin Groden, he stepped in, and also I have to thank Michelle for stepping in because we lost my co-chair who was a cardiology lead here. So, but anyway, this is our group of folks looking at various different endpoints. And so obviously for endpoints, you know, the goals, accelerate the development of evidence and knowledge to optimize the selection, use, and evaluation in clinical trials for drug developers and from evaluator standpoints. And so, again, we heard a lot about endpoints in the prognostic session, which is really important and great work. And so I'm glad that we had some of those discussions already, and we can certainly continue them. But obviously, endpoints are essential for clinical trial design. And this forum is a place where we can kind of really try to nail it down a little bit. And there has been regulatory success in the heart and nerve area in amyloid. But again, as patients are now diagnosed earlier, we have to really reconsider what our endpoints are, because things like, you know, death and hospitalizations and things like that and 6-minute walk may not be adequate. So, and again, there are a number of different approaches that we can apply. One of the real questions is how much do we have to separate out endpoints when you're looking at organ-based things? How much do we have to separate out for AL from ATTR? I would say that, you know, AL cardiomyopathy is different than ATTR cardiomyopathy, and so though we might use the same tool, how you use it and what the endpoints might be would be potentially different. And then there are just some things that are just specific to the different diseases. So we're going to pretty much at this point limit our conversations to a few areas. But again, as far as the definition of endpoints is a targeted outcome of clinical trial that's statistically analyzed to help determine the efficacy and safety of the therapy being studied. And an endpoint may include one or more clinical outcome assessments and or surrogate endpoints. Clinical outcomes assessments measure direct clinical benefit to the patient. Surrogates, including biomarkers, predict clinical benefit. But obviously, we have to validate. So no matter what we're talking about, we need validation. And so, you know, this is the amyloid space of trials, like phase 3 trials. And we only have one success story there, and that would be the Andromeda trial, newly diagnosed AL, MOD-PFS, which was an endpoint, which was a creative endpoint. And there are two that are at the bottom that haven't read out yet. But there are different endpoints that have been used in the AL space, but we only have one approval. The ATTR space, a little bit more success and perhaps more success to come. And again, you can see the various endpoints. Complicated neurologic endpoints, the the MNIS+7, which is a rather onerous kind of endpoint. It has gotten us approval, but moving forward, and if we want to have trials that may include cardiac and renal cardiac and nerve patients in the ATTR variant space, it really does get very tricky as far as picking centers and all the rest. And again, as I mentioned, in terms of the cardiac endpoints, perhaps we need to, something like a CPET may be a dream of the future. And so the goals really, we want to sort of simplify, validate these different endpoints. There's more and more heterogeneity in trial endpoint design, so we need to really carefully. The funny part is AL has been around longer in terms of, doing little studies, this, that, and the other, and we really don't have as good endpoints in truth or accepted good endpoints as do the ATTR in the ATR space. And so that's work that needs to be done, and we're going to focus on that in one of our sessions. And then I already said this about as patients live longer, some of the CV death and hospitalization has to be revised, and simplifying the neurology endpoints. And then there are just new things, new strategies like minimal residual disease in AL, which hasn't been validated, but is certainly something that people think of, the various imaging modalities, and other markers of peripheral nerve involvement. And so this session, we will start with the hematologic endpoint proposal, PRO endpoint proposal, renal endpoint proposal, and then there's a lot of time for discussion. And so with that, we'll move to the first talk, which is mine. And so this one is, I'm not going to give tons and tons of background. This is kind of a call to action. We've talked a lot about how do we change endpoints, what are the shortcomings and so forth. And so trying to really roll up my sleeves a little bit and see what we can come to agreement with. This is our group. And so the two biggest challenges in hematology endpoints are what we call hematologic progression. So using the 100 mg per liter or 10 mg per deciliter is not a tenable endpoint anymore. Nobody wants to wait that long. And then the other major challenge is the concern about what measurable disease is when it comes to enrolling a patient. So the eligibility, the minimum amount of light chains do you need to get a hematologic response? And, there's some tweaks that could happen in terms of, CR and do we include MRD? These are all important things, but these are two very important pieces. And so that's what I'm going to focus on in terms of, an objective, and this is going to get into the hope for collaboration and using federated analytics and having buy-in from, industry to share data and so forth so we can really figure out where we are. And so, again, to use data sets to test proposed alternative definitions for hematologic progression. There have been different conversations and different definitions that have been proposed or talked about, and we'll go over that briefly. And then also in terms of the measurable disease thing. So entering patients with what we call a DFLC or a difference between free light chain of 50 mg per liter. That was the downfall of part of the downfall of the Torlamain study because people didn't want to wait that long to put patients on, and so it was a disincentive. And then there's conversations, well, should we use 20 mg per liter as an eligibility criterion? And the question is we don't really fully understand the variability of the free light chain assay, especially for people who are on therapy and such. And so the question is, can we use existing data to get a sense of what is the wiggle, before we commit to something that we may regret. We want to really understand the behavior. And so, again, the first point is looking at progression. And so if we imagine a few scenarios, so one scenario is you have gradual light chain increase without meeting the 2004 hematology progression definition, and the patient is just taken off the study. And so, again, what happens here is you have time, the patient is essentially a sensor for progression. If you're looking at time to next therapy, sure, you get that point there. But if you had a new definition that people could live with, then you could actually capture that event and not just sort of throw that patient away in terms of your event, because a lot of times studies are maybe powered to progression events, and if you censor your patient, you don't get an event. And so by bringing it into reality and what providers are comfortable with, that is a potential advantage. Another scenario is there's a gradual increase in the light chain. Patient remains on study until the full heme progression criteria event met. In the U.S., that doesn't happen very much because we have access to a lot of drugs. In other countries, sometimes they don't have access, so they will keep patients on. So what would it do? You have the PFS event on the timeline. If we had a new PFS definition, it would basically shorten the time to PFS. And so that, in a way, also is a good thing in terms of just sort of getting to an answer sooner. Obviously, scenario 3, where a patient doesn't have an adequate hematologic response and the patient's taken off because of that reason, we won't be able to, that changing progression won't change that. That's just a trial design thing. But just sort of getting a sense of where the importance lies in thinking about having a more reasonable PFS definition. So this is kind of busy, and the gist of it here is on the left-hand side, you see 2005 consensus, 2012 consensus about what the definitions for progression are. And essentially, they're as such, they haven't changed, but the key is that you need to get your DFLC up to a minimum of 100 mg per liter. Back in the day, in 2005, we didn't have good therapies, and so this was a very reasonable endpoint. And so after a lot of conversation and review of the literature and all these other things, there are some in the sort of, if you look in the progression, kind of in the middle, in the kind of bluish-green, whatever color that looks like, here is a variation on the high-risk progression of Palladini that was published 6 years ago. And basically using the absolute value of a DFLC of 20 mg per liter, 50% increase from the level at best response. There also is a little tweaking that could occur to really clean up this relapse from complete response is also shown there. But again, these are things that could easily be tested in data sets, but we don't want to commit to something before we know how it really works in real life. And so again, here is kind of using examples, little spreadsheet, which I'm not going to go through, but the idea, the thought experiment is if you start here and you go here, what happens? When we talk about some of these definitions, we have to be careful because, again, if we talk about changes, a delta of, best response to progression, do we trust the assay enough that that's going to work? Because you can't have it both ways. And so that's where using federated analytics and using existing data, I think we can explore these kinds of things. And so, in whom would we try this? Again, there would be patients enrolled in different treatment trials that have monthly measurements of the monoclonal protein. They would have had to have had clone-directed therapy, newly diagnosed or relapsed. A high level how is just looking at the various opportunities of progression criteria and see how those work and benchmark those to things like, the MOD PFS, if available, recalculate and benchmark it to overall survival and organ improvement and do sensitivity analyses. And obviously the data needed would be from existing phase 3 trials. And so if those were available, we would have reasonable ends to really make a good design. In terms of, the elements needed, again, here, they're the usual suspects of just a lot of labs, and things like that to know, and some endpoints in terms of survival. And if the raw data are there, a lot of things can just be calculated. In terms of then really objective 2, which is really similar to objective 1 in the sense it's really looking at the variability of the assay. And so, low levels, could we trust a 20 mg per liter? And then how does, if we start somebody on a study at that level, what does that mean in terms of how we judge response and progression? And so, as I mentioned, this was a major, or one of the major issues for the Torlamaine AL trial not succeeding. It ended early. For newly diagnosed patients, it's less of an issue, but in the relapse setting, this is more of an issue. And so, again, if you start with such a low level of free light chain, your only response grouping that you're eligible for is a complete response. MRD is another type of response, true, but that hasn't been validated, so that doesn't get us to trial development and endpoint at the moment. And so that is something. So the variability, the performance of existing or modified response criteria at this low level, what does it look like? Let's understand it before we actually commit. And so way back, we did I guess it's 13 years ago, we looked at biologic variation of the various assays, and, the serum free light chain is like a 28% variability, but it wasn't checked at such low levels. And so, again, one can look at these things, but we really need to know in modern day with low levels. And so, again, the on-study free light chain is really intimately related to response. And again, in the setting, as we're looking at the variability, the thing here is that we would basically say, let's test the response criteria as they are in terms of, MRD hasn't been addressed to this point. Certainly, if there are data, we'd love to look at that. But also really looking at getting rid of the free light chain, perhaps, the free light chain ratio in CR, and using a modification of existing published criteria, and then also looking at, let's see, what else do I have here? Oh, right, and re-looking at what partial response might mean, and just there's a lot of little mathematical kind of looking at it. And then the other question is, how much is bone marrow needed in the, in the CR category? If we have an MRD negative category, perhaps we can keep the bone marrow out of CR and leave it as it is, and then just have another category. And so I'm going to just, again, this idea of, do we trust the variability? Are we going to trust a change of 11 mg per liter to say that that's an improvement, a worsening, and all the rest? And so in sort of the objective, we're going to validate the variability. We also will track the immunofixation in studies to see how often positive, negative, like what is the actual performance of these tests on these trials. And again, we just need patients that have been on clinical trials, and the same trials as before, some basic demographics and laboratory, and then, this is sort of a non-statistician's thought on how one might look at these various definitions of variability, looking at it at different time points, at different levels, and then understand what we're dealing with and what we can use as our definitions. And the same thing with FIX, and then this all can be done, as a calculation, a computer-assigned calculation if the data are supplied, and we can test different combinations and permutations of CR and relapse from CR. And again, all sorts of little variations have been used and described, and the goal is to really nail it down. And so with that, kind of looking at this grid of, what are some new things to look at, what are potential definitions so that we can really move things forward, stop talking about it, but actually do something about it in good quality data, I think that we have an opportunity. And so what would a timeline for this kind of proposal be? I mean, if we can get buy-in and we can get data, the data would already be available, and the project can move forward as quickly as resource allocation permits. There is a survey that is sponsored by the ARC looking at practice patterns and so forth that we'll be distributing soon, and we'll get some information from that. But again, it's a question of prioritizing that this is an important thing to look at, sponsor sign-on, survey of the experts, like I said, so we can really, say, okay, do we have it right, or what are the different kinds of endpoints we want to check, finalize the statistical path and the endpoints, clarify those data points, the federated analytics, review and write and move the field forward. In terms of challenges, getting the data is certainly going to be a challenge, harmonizing the data. But I think that this forum is a great opportunity for sponsors to collaborate and for us to actually answer questions. Again, the harmonization of the data, making sure we know fields are fields, but if it's federated, we should be able to clarify and explain all of that. And then might need some creative statistical analyses and so forth, but again, that's what we get talented statisticians for. So the questions that I would pose to you all, the nuance of endpoints, yes, what do you think, and what makes sense as far as industry, same question, and please engage. Regulatory, would this affect change? And patients, obviously, we always value your comments and input. So that is all I have, and I think we go. Yeah, so with that, I will invite Dr. Vaishali Sancharwala to talk about KCCQ in AL clinical trials. So, Vaishali, thank you.
So good afternoon, everyone, and thank you, ARC, for organizing this wonderful event. This is my 5th one, I think. I've been here since the first one, and it just gets better. Every single forum has been just phenomenal. So thank you very much. I am Vaishali Sancharwala. I am part of the Endpoints working group, and my job is as the chairperson for PRO subgroup of the Endpoints working group is to propose a project. And on behalf of my co-chair, Michelle Carty, and myself and Avery Rizio from Quality Metric, we have come up with this proposal of incorporating KCCQ in AL amyloidosis clinical trial endpoints. And the main objectives of this project are to identify PRO instruments that have been used to evaluate health-related quality of life among patients with ATTR amyloidosis as a background. And then the second objective is to identify, based on existing recommendations, a PRO instrument that may be suitable for use in patients with cardiac involvement across both AL and ATTR amyloidosis. And finally, to use FDA guidelines to identify and fill gaps in the existing evidence to support the use of instrument in PRO-based endpoints for clinical trials. Today, this talk is going to outline the work that we have conducted so far and outline the next steps to meet this objective. So obviously, this is not a completed project. The signs and symptoms experienced by patients with AL amyloidosis are largely driven by which organs are affected. And this was demonstrated in the updated conceptual model demonstrated here on the right that we published in 2023. Patients with cardiac involvement experience, and again, cardiac involvement is present in 70 to 80% of patients with systemic AL amyloidosis, and patients experience fatigue, weakness, shortness of breath, swelling of their legs, among many other symptoms. And the clinical trials designed to evaluate treatment efficacy among patients with AL amyloidosis and cardiac involvement include most of the times the biomarkers, specifically NT-proBNP. However, it is critical to evaluate treatment efficacy in terms of how the patient feels and functions. And identifying a patient-reported outcome measure that can capture treatment-related change in cardiac symptoms and impacts is an essential component of AL amyloidosis clinical trial measurement strategy. We at the Amyloidosis Forum in 2021 were part of the Health-Related Quality of Life Working Group. And this working group published a report called Health-Related Quality of Life Instruments for Clinical Trials in AL Amyloidosis. This report was published in 2023, and we concluded in this report that measuring outcomes relevant to patients, such as health-related quality of life, should be recognized as a critical component of drug development in AL amyloidosis. Moreover, we also suggested that SF-36 version 2 and Promise 29 were identified as instruments relevant to patients with AL amyloidosis, and these have demonstrated evidence of reliability and validity. And as a result, these instruments should be considered for inclusion in clinical trials with this patient population. In 2024, earlier this year, Anita D'Souza from Medical College of Wisconsin published this paper on development and initial validation of AL profile as a PRO instrument in patients with AL amyloidosis. It is important to note that this was a combined instrument that had selected items from Promise 29 and PRO CTCAE. This paper also suggested that there was content validity by debriefing interviews in 20 patients with AL amyloidosis, and 297 patients completed AL profile side by side with SF-36. And what it demonstrated was there was a large correlation with reliability and validity. And the conclusion of this paper was that the AL profile has acceptable reliability and validity for use in systemic AL amyloidosis. However, this has not been studied prospectively in clinical trials. Now, KCCQ, Kansas City Cardiomyopathy Questionnaire. So it's a 23-item PRO instrument that measures health status over the past 2 weeks in patients with heart failure. 6 scale scores and 2 summary scores are produced, where higher scores indicate better health status. The 6 scale scores are symptoms, physical limitations, quality of life, social limitations, self-efficacy, and symptom stability. 2 summary scores that are part of KCCQ is clinical summary score and overall summary score. A short version, KCCQ 12, includes 12 items that produce 4 scale scores and a single summary score, which integrates all 4 scales. In 2016, the FDA qualified the symptom, physical limitation, social limitation, quality of life, and overall summary scores of KCCQ for use in quantifying patients' health status due to congestive heart failure in medical device studies. In 2020, the FDA qualified total symptom, physical limitation, and clinical summary score of KCCQ 23 as measures for use in drug development in studies with congestive heart failure. Though not developed for patients with AL amyloidosis, working groups focused on recommending relevant endpoints related to both health-related quality of life and cardiac outcomes have identified that KCCQ as an instrument that warrants further investigation and consideration for use in clinical trials of patients with AL amyloidosis and cardiac involvement. Again, this is from Amyloidosis Forum 2021, and cardiac working group led by Dr. Maurer proposed some cardiac endpoints for clinical trials in AL amyloidosis. And the prioritized cardiac endpoints that were suggested in this report were survival, hospitalization, biomarker NT-proBNP, 6-minute walk test as a functional test, KCCQ, and cardiac progression-free survival. Focusing on this, we also noted in this paper that KCCQ was not validated and had no natural history study in patients with AL amyloidosis. Meaningful threshold was defined for KCCQ, however, for patients with congestive heart failure and not particularly for AL amyloidosis. This is a summary slide of KCCQ in clinical trials for AL amyloidosis from clinicaltrials.gov. There are 2 clinical trials for AL amyloidosis incorporating KCCQ as secondary outcome in prospective clinical trials. However, there are 20 clinical trials that were identified on this which have incorporated KCCQ in ATTR amyloidosis clinical trials. The AL amyloidosis clinical trials that have incorporated KCCQ as a secondary outcome were KALM 301 and KALM 302 trials. These trials have completed accrual but have not been analyzed yet. And the KCCQ-based secondary outcome was changed from baseline to week 50 in overall score of KCCQ 23. These are the trials that you have seen initially also in other slides, where these are the key clinical trials for ATTR amyloidosis. And these first 3 trials have completed accrual, and we have top-line results of these trials where KCCQ-23 was used as a secondary outcome measure as the PRO instrument, with overall summary score as the measure or the score that was evaluated in these trials. The next 3 clinical trials are ongoing and are also utilizing KCCQ-23 with overall summary score as the secondary outcome measure. It is important to note that inclusion as a secondary endpoint in a trial does not mean that it has been accepted as an endpoint by the regulatory authorities. KCCQ labeling for tafamidis for treatment of cardiomyopathy for ATTR wild type and ATTR variant amyloidosis was first done by FDA in 2019. And a significant treatment effect favoring tafamidis was first observed at month 6 and remained consistent through month 30 in KCCQ overall summary scores. Now, inclusion of KCCQ in clinical trials for AL amyloidosis with cardiac involvement represents a promising step forward in measuring health-related quality of life from patients' perspective. However, simply demonstrating treatment differences on this outcome is not sufficient to support regulatory decisions and inclusion of labeling language. The U.S. FDA requires that sponsors provide evidence that a selected clinical outcome assessment, such as KCCQ, is fit for purpose in the trial-specific context of use. And publicly available evidence can be used to support the use of a clinical outcome assessment, and collecting and publishing this evidence as it pertains to KCCQ can help support wider spread use of this instrument in clinical trials in AL amyloidosis with cardiac involvement. So what evidence is available and what is still needed? With respect to content validity, a more thorough evaluation of the symptoms and impacts experienced by patients with AL amyloidosis and cardiac involvement is needed. The way in which KCCQ evaluates concepts relevant to patients with AL amyloidosis with cardiac involvement is also needed. Data from patients with AL amyloidosis and cardiac involvement to demonstrate that they interpret the questions and response options as intended and can select a response that accurately describes their experience. With respect to reliability and construct validity, what is needed is a comprehensive psychometric evaluation of KCCQ among patients with AL amyloidosis and cardiac involvement. Data for these analyses could be derived from an observational study and/or clinical trial data. Analysis must follow a psychometric analysis plan that outlines a priori hypothesis for construct validity in addition to thresholds for interpretation of results. With respect to ability to detect change, a psychometric analysis that demonstrates that changes in KCCQ scores are associated with changes in patients' health status as reflected by other outcomes is definitely needed. Quantitative analysis to estimate meaningful within a patient change thresholds and meaningful score differences that can be used to interpret change on the KCCQ that is observed during a clinical trial. While such analyses have been conducted in other patient populations, specifically patients with congestive heart failure, the evidence must be generated using data from patients with AL amyloidosis and cardiac involvement. And this is the roadmap for the next steps for this working group. We need to do concept elicitation interviews, cognitive debriefing interviews, psychometric validation and interpretation thresholds, and finally, dissemination by publication of the information, which will allow sponsors to use the findings when appropriate to support regulatory submissions. Thank you very much for your attention, and thank you for this opportunity.
That was wonderful, Vaishali. So we're going to keep all the questions towards the end. So we have one more talk, and that one's online. So Dr. from Mayo is going to talk about renal response and progression in AL amyloid, examining the interaction between the change in proteinuria and the change in eGFR.
Okay, thank you, Angela. Do you hear me?
Yes.
Okay, good. I'm sorry that I wasn't able to come. I wish I could, but I couldn't. So I'm presenting this proposal on behalf of the Renal Endpoint group. I assume that somebody will move the slides. Thank you. So the basis for our proposal for looking at the interaction or the changes in eGFR and proteinuria are at the footstep of work that has been done recently and prior. So in renal AL amyloidosis, one of the major components of the disease manifestation is typically proteinuria. And this is usually what is responding eventually, if the treatment is successful, in terms of the kidney outcome. Proteinuria is coming down, and then we know that the patient is responding to the treatment. Until recently, we had a binary response criteria that were formulated 20 years ago and 10 years ago were revised. And we came with a proposal to look into if we grade the renal response from a binary to a 4 level, if that will make any difference in terms of prediction of renal outcomes. So on the left hand side, you can see the a general view of those 4 response criteria for the kidneys in AL, and it goes from no response when there is less than 30% reduction in the proteinuria from baseline to complete response, which is not measured by percentage reduction like the other 3, by reaching a threshold which is considered a normal threshold for patients without renal AL amyloidosis. And as you know, those responses do take time. We give the hematological therapy for patients, but the observation of response does take time, and this is one of the challenges with this disease. So by 6 months, for example, we have less than 50% or close to 50% of patients having any type of renal response, but that significantly increased over time. And in 24 months, more than 70% have some form of renal response. And also with time, we see deeper and deeper renal responses. and more than 20% of patients actually can reach a complete response as their best response, which is not even at 24 months. Sometimes it takes even longer than that. So that does take time. Next slide, please. And then we correlated that with renal survival and overall survival. And this is one of the things that kind of, I think is important message from this study is we can see that on the top panel is progression to dialysis. So the higher the graph goes, the more patients go on dialysis. And you can appreciate that the better, the deeper the renal response, the less likely the patients will progress into dialysis. But on the lower panel, this is for overall survival, and especially if you focus on the 24 months time point, people that get into renal very good partial response or renal CR actually have a survival advantage over those that have a lower depth of renal response. So, so there is a survival gain by getting into a deeper renal response. Next slide, please. But one of the challenges is that while we can measure proteinuria and understand the degree of renal response, when we try to look at renal progression and try to do that with proteinuria, any degree of rise in the 24 urine by itself did not have any impact on renal survival. However, and we know that from prior works, a change in eGFR, in this case, this is traditional metric, which is quite stringent, more than 25% increase in eGFR from the nadir value does correlate with renal outcomes. So this is at 6 and 12 months on the left-hand side. You can see people that have eGFR progression by this definition at those time points were more likely to progress into dialysis. And that also translates into an overall survival difference. And we know that dialysis is generally not a very favorable outcome for patients. So next slide. So we don't have really any good data, follow-up data, that shows the interactions between the proteinuria and the eGFR. And if we can have this assessment, longitudinal assessment of both the proteinuria change and the change in eGFR over time, first we can improve our understanding about how is renal response and progression kinetics look like. We can explore optimal cutoffs for renal response and probably for renal progression. Again, 25% is quite a stringent criteria. It will allow us to identify time points in the journey of the patient that actually, if we intervene successfully, we can reverse bad renal outcomes. And so we can use that. And we hope that with prior works and this type of work, we can actually suggest to the regulatory authorities that renal biomarkers, the 24-hour urine protein and the eGFR, are relevant endpoints for renal AL amyloidosis. Next slide. So generally, what we have been discussing is to perform a multi-center study, consortiums of centers would treat patients with major centers treat patients with amyloidosis across the globe to investigate the change in eGFR and proteinuria over a course of 3 years from the time patients started their treatment. And we would hope to validate this data with data coming from clinical trials in patients that are newly diagnosed and have renal amyloidosis. Next slide, please. For a little bit more specific aim, so aim number 1 is I just discussed changes in eGFR and proteinuria over the course of 3 years, but I think it's also in the era that we are using daratumumab as our first-line treatment, it will be good. What is the differential impact of daratumumab versus non-daratumumab treatment on these renal measurements and also renal outcomes? I think this is another benefit that we can learn from such study. Next slide. So this is the detail. So what we want to do is to do a multi-center study that will include AL patients who were diagnosed between 2017, beginning of 2017 to end of 2022. This is a 6-year period. We anticipate that approximately 30% of patients would have received daratumumab-containing regimen. It was approved in 2021. It's available in the US from 2021, but some people got it even before. So maybe around 30% will be DARA patients. Obviously, patients need to be accessible for renal response. We'll take patients with at least 0.5 gram of proteinuria, mostly albuminuria. We can then do subgroups for higher threshold. And patients must not be on dialysis time of diagnosis, because then we don't have any renal measurement to document. We want to take patients that have hematological partial response or better within 12 months of treatment initiation. This is irrespective of how many lines need to be given to reach that goal, because patients who do not have a response are unlikely to have renal response, and it's kind of making the work a little bit easier. As far as what do we want? So obviously we'll have some baseline information, treatment data, but what we want is have a serial measurement of both creatinine, from which we'll calculate the eGFR, as well as 24 urine protein. And we want to do it serially, initially, especially during treatment for the monthly for the first 6 months, but then every 3 months, and then eventually every 6 months. And that will culminate, I think, into 15 time points, and also include best renal response. It's important to recognize that we want to include patients who will not have all data in all time points, and I assume that at least in a retrospective study, that will be at least some of the patients, although those are measurements, are typically measured for those patients, especially creatinine, maybe less than 24 urine protein. Data sources will be the electronic charts of patients in those participating centers, and a validation data set from clinical trials in newly diagnosed AL patients, and some of the trials that are either completed or still ongoing. Next slide. So we plan to engage previous centers that were engaged in the previous work on renal response, and we want to invite few more centers, so we have a little bit more representation worldwide, and also increase our power, as with our better treatments, it might hard to get to a hard endpoint such as death or progression to dialysis. We hope to start the collection sometimes in the beginning of next year. We already established the data collection sheets with us and the protocol. So hopefully, after engaging with centers, we can start the data collection next year. Hopefully, within a year and a half, we can able to collect all the data. Since we are enrolling patients that were treated as late as 2022, this will allow us at least 3 years of follow-up for patients, because this is what we want for all patients. Obviously, if they made that landmark. Data analysis will be the following year. I do assume that it will take time. So it's a little bit more loose here, 2026, 2027. And hopefully we're able to move it out to a publication, so and that may help disseminate this information sometimes in mid to end of 2027. I think I have one more slide, maybe. Yeah, and this is kind of, we basically we want feedback to see what people think about this idea, if they have any ideas for improvement, getting some validation data from industry. I hear from regulatory about how likely to accept those renal measurements as a valid endpoint in renal AL. And obviously, we are also looking for feedback and comments from patients. Thank you very much.
Thank you very much. So it doesn't look like there are any questions on the chat there. I would invite my co-chair to maybe make some comments about things, perhaps.
Thank you. We're very excited in this session to really make this more of a working group. We have these ideas about what we'd like to move forward, and we feel this is such a great opportunity to collaborate and hear, well, you know, what are your concerns? What are the things that we need to include in these plans? One of the things that's really great about having the Amyloidosis Forum is that you can see there are some ties going across each one of these ideas, and that the fact that we are collaborating together allows us to have less burden on a patient. I'm seeing some of the ideas that other work groups have, other than the one I'm in with patient-reported, and I'm thinking, oh, I would like to help think about what measures some of the data that's being collected or used from centers in those ideas are, and how I can help ensure we're tying those things in and learning what we can learn. When talking and thinking about Vaishali's presentation, just going back to one of the first slides, we had just wanted to say that we were building on what's been done before. So as she was presenting about work in a systematic literature review done on health-related quality of life measures in AL amyloidosis, we do have a couple different ideas moving forward, and one was the first kind of thing she talked about of, well, we should really replicate that in ATTR. Back in 2019, when we had this meeting, we didn't have any disease-specific measures that were reported by the patient, but now we do. We didn't have, you know, much evidence at all out there, and now we have more. So one thing that could contribute to the field is to conduct a similar literature review like we've done in AL, in ATTR, and make sure that then everyone who's doing research in this area has that information available moving forward. One of the other things that I wanted to talk about is what we were showing there with the KCCQ is that, hey, we know people are using it, and what is there going on? There's what we know, what we can show, which each individual company might have to replicate and do the same work over and over again, and then what we can collaborate and get published out there so that we make it easier and make it faster to get this research done and out there. You know, seminal work by Wilson and Cleary in 1995 discussed the four-box model of concepts that are important to measure from what's proximal to distal in a trial, and patient-reported measures are often not the closest thing that you see change or be modifiable by a treatment. However, they are very important. And in the last 20 years, both NIH and FDA have made great advances in supporting patient-focused drug development, which is a great part of this public-private partnership we have. And in that, they started with a draft in 2006 that was then finalized in 2009 of really the rules around using patient-reported outcome measures as endpoints in clinical trials for medical product development, and that has now morphed into 4 separate guidances, 2 of which are final and 2 are drafts, that kind of lay this out. And so what we were showing there with the KCCQ is, hey, we know that the KCCQ has been used in all these things, but it's not really out there in the literature in all of the evidence that needs to be shown if you're going to have a dossier to support that as an endpoint. Part of the initial presentation was showing primary endpoints, but I think our second presentation was showing that there are also considerations for secondary endpoints and exploratory endpoints, and that's really where some of the patient-reported outcomes can come from. Some of the things that are missing as well is evidence of meaningful change and a good, solid definition of what the endpoint is. So we've talked a little bit here about a measure, the KCCQ in our case, and then some others, obviously. And then we've talked about what those scores are that come out of it. But what makes that an endpoint? And is that something that can be modifiable? And should that be something that you consider for all trials, for certain types, and going back to that fit-for-purpose evidence that we have to think about. So I just wanted to put that out there, and also for everyone, expanding beyond regulatory. We talked a lot here with endpoints that we are talking about regulatory, but some of what we're doing needs to get further put out to payers and HTA groups and non-U.S. as well. So I'm very excited to hear what thoughts people have and what questions they have.
So I'm going to give Dr. Groden a chance if he wants, and also to Dr. Kella, both of you, a chance to just any thoughts on cardiac and neuro. Again, we know that you didn't have time for a proposal. These things were last minute, and thank you for engaging. But please, I'll start with Dr. Groden.
Thanks. And this has been a terrific meeting. I'm sorry to not be there. I think, and I'll be brief because I think we've got a number of topics to discuss, especially on the heels of what was just discussed a few moments ago by my colleague. I do have one comment on that, and I think it is a slippery slope when we're dealing with payers and what we define as response, because sometimes that can then backfire on the patients themselves if we as clinicians are not quantifying it, or if we're not capturing it, perhaps the same way, that they would intend. Sometimes, therapies might be withdrawn or funding for therapies might not be approved. And then from a cardiac standpoint, I think there've been a number of excellent presentations today. And a lot of what we focused on does overlap with, some of the surrogate and perhaps non-amyloid, but yet, or non-amyloid target organ disease endpoints. We focused on PROs, and, I think when we met with our group, we were trying to come up with endpoints that we would suspect or hypothesize might be, one, responsive to therapy, and two, track with prognosis, and if modified, would be related to the effect of the drug on outcomes. So thinking of things that really might reflect the Prentice criterion. As you all know, that's quite a high bar, but we did touch on, and I know this was discussed this morning, on AI metrics, not necessarily for the assistance with imaging, but more so as a phenotype in of themselves. So I know many of you in the room are familiar with echocardiographic-based AI metrics. There's an industry company there's a company that is developing one such algorithm. I'm sure there are more. I know of some academic centers that are also developing this, and also ECGs. So, these are largely used for disease or case finding in a population at risk or that might be enriched for risk. But I think one thing that's very interesting is that these diseases or these AI algorithms are in essence giving us a risk score for something that the algorithm is detecting graphically. And whether or not that might be an amyloid phenotype would need to be validated. So, we did discuss those as two potentials. Obviously, it's not as well formed of a hypothesis and as detailed as Angela and Vaishali have presented to you this afternoon. And then another was more PRO based, and this is probably more specific for ATTR, but we were thinking about a PRO that the NIH recognizes as important, the SPPB. Again, this is most of our patients are older, and so very gerocentric outcomes seem to be quite important and patient-centric in these individuals. Now, whether they're modified by therapies or not is unclear. And then the last thing that we we're discussing, and I think all of these potential endpoints that we were attempting to come up with, we really tried to focus on feasibility of data that were already collected so that, we weren't having to do something prospective. So another was taking this concept of home time, which is the time individuals spend not only outside of the hospital, but outside of an institution, such as a rehab facility or a long-term acute care facility, et cetera, that really is a patient-centered outcome and then could be derived from clinical trial data, because I would assume that they're keeping track in, as to where these patients are being disposed once they have been hospitalized. And this is probably more important for those that are sicker or more dependent or more frail. So there's a lot of fleshing out that needs to happen with those ideas, but those at least 4 topics should give everyone in the room an idea of what we're thinking about.
Thank you. Dr. Kella.
Sure, thank you. So from a neurologic standpoint, I think one of the things that maybe was highlighted briefly today was the neurofilaments. And measuring neurofilaments may be an outcome that needs to be defined better and followed more carefully. And this would be a little bit more objective than the neuropathy impairment score, which is clunky and somewhat, and in part, a lot of it is not really relevant to quality of life. And so I'll just keep my comments, because in the interest of time, brief, but I think that would be sort of an approach.
Thank you. So I have a specific question for Vaishali. So you talked about doing kind of like the focus group thing with the KCC score. I mean, is it needed because it's already such a validate? Like, I get it, like if you're modifying something or help me understand, like, please.
So it is not truly, quote unquote, validated for AL for sure. I think what we did with SF36 version 2 or SF36 on the whole for AL when we proposed it was there was all these steps that were done appropriately, like content validation, psychometric property validation, debriefing interviews. And then we said, yes, this was also studied with organ-specific responses that SF36 measures did improve. It was all based on observational studies as well as based on debriefing interviews from patients. And obviously that was mainly done with, my friend Spencer is in there and Michelle's group. I think my question before Michelle answers your question is that, in KCCQ, I recommended or I suggested that we could do psychometric validation based on observational studies, but I don't have that data. So KCCQ data is being collected for ATTR amyloidosis, and it was collected only on 2 trials with KALEM. And is KALEM the right clinical trial to study because we are using an antifibril agent as well for KCCQ? I don't know. And that was the question for Michelle, that how are we supposed to do psychometric validation because we don't have the SF36 data that we had for AL amyloidosis. Other centers might have KCCQ for AL, but, I don't.
Yeah, and one of the slides that we had was showing a box on the right that showed all of the evidence, well, not all of the evidence, but a summary of the evidence that is needed. If you're going to use an endpoint in a clinical trial, and that's what we're talking about here is endpoints for a clinical trial, the FDA is going to want to see evidence of all of those particular needs for validation, as you mentioned, content validity, psychometric validity, responsiveness to change. And there are very many different parts of that you show with each. And one thing that we found when we were looking in the literature is that, hey, well, I'm sure that many, many patients have taken and used the KCCQ and AL. There is not a publication showing that the specific recommended methods that you would use interviewing patients to make sure that they find the measure to be representative of their experience, that it is comprehensible, that it is comprehensive. Those are the things that we have to show, and that's why doing a study like that, which is relatively low-hanging fruit. It doesn't take that much effort to be able to do, and publishing that as a group, that means every group that goes out and wants to use that would then have that available to use as a reference and to put in their materials that they submit in a dossier and an NDA submission for FDA. And so that is just one of the things that you have to have. You also have to have your really full conceptual framework with your conceptual model, your measurement model, and showing how all these things are combined. And that was one of the things I was mentioning, is that you don't just need to say the KCCQ. Which score is it, and how are you framing it? Change from this to this on this particular score is anticipated to be changed or modified by this treatment. And all of that has to be kind of put together with the evidence of the validity of the measure in all those ways. And so I'm going on and on, but that's what I was saying.
So we haven't heard anything from any of our FDA colleagues. I don't know if there's anybody in the room who wants to reflect back on anything that has been said.
Yeah, thank you for the great presentations. I do have a question. Are you trying, so KCCQ or some aspects of it have been validated for the heart failure population. Are you looking at your proposed population that does not have heart failure? Or do you think, are there distinct characteristics of the population that you're trying to study whereby you wouldn't expect KCCQ to perform as it has performed in heart failure populations?
So, you know, we are looking at cardiac involvement from AL or ATTR, you know, and depending on their stage, whether it is, you know, early stage versus advanced stage, the congestive heart failure could be different. NYHA class could be different. So I think that, you know, whether this particular instrument performs in those patients or not. So that is, and, you know, it has been studied in multiple trials for ATTR. So we have prospective data on that, and now, you know, should we do it in AL?
Thank you. So I just wanted to add that symptomatic heart failure patients, if that's the target population, and you wanted to propose KCCQ as one of the endpoints, I don't think we'll be averse to that. If you are trying to look at some metric of quality of life in a population that probably has some type of cardiac involvement and other manifestations of the disease and is not quite symptomatic heart failure, then I wonder if there are maybe other PROs that may be more appropriate. I do want to share some of the challenges we face in interpretation of KCCQ, and you've pointed to some of those issues as to what is the minimum within patient change that would be perceived as an improvement. KCCQ is a composite endpoint of sorts, and so it does suffer from the issues that we would see in a composite where maybe one domain or item is trending in some favorable direction, but other items haven't moved or maybe have moved in a different direction. And, you know, how do you make sense of it? And then are you missing a real treatment effect just because you used this metric? We've also sort of recommended to maybe consider collection of anchors within the trial that's evaluating KCCQ, so we can try and use those anchors to understand whether or not the treatment effect that was observed is actually clinically meaningful, unless the KCCQ change was large enough where we are not debating that there is a benefit that's been observed. So I just wanted to add that.
Thank you.
Thank you. Very helpful.
Can I ask a question? Oh, I'm sorry.
I think.
Well, I'll let you ask, because I was actually going to comment on Dr. Mukhtar's presentation. So do you want to keep going with KCCQ? Okay, great. Excellent timing then. Hi, my name's Elisa Thompson. I work with Charu and Norman. And I just wanted to say this has been I've learned a lot from this meeting. It's really been a great discussion. It seems like a wonderful group. I just wanted to maybe talk for a second about the kidney endpoint issue, and really make just two sort of high-level comments about surrogate endpoints. And I think the first comment is more about the evidence supporting the surrogate endpoint, Dr. Mukhtar, and then the other is sort of how we think about what constitutes sort of, the amount of change you'd want to see in the surrogate to know that you've done a good thing, for lack of a better term. So generally speaking, and you probably know this, and all in the room probably know this, but generally speaking, when we think about surrogate endpoints and the body of evidence supporting them, there are three buckets there. One is really that scientific rationale, understanding of where the surrogate sits on the causative pathway, or this putative surrogate sits, right? Then there's this sort of bucket of sort of data, which is sort of what you're pointing to, which is the associational data, right? How does my surrogate at baseline or changes in my surrogate over time track with the outcome, in this case, kidney failure? And then, finally, that best bucket of evidence is the evidence from randomized controlled trials showing that, various treatments that work through different sort of mechanisms of action, that treatment effect on the surrogate reliably predicts the treatment effect on the outcome. I would say in many of our diseases, we actually don't have that third bucket. And so to the extent that we can arrive at the conclusion something is a validated surrogate, it rests very heavily on not just the associational data being incredibly strong, robust, consistent, but really a clear understanding of the causative pathway and where the surrogate sits on that. So it sounds like, for you guys, you're really sitting in the space of, we're going to try to come forward with the validated surrogate, but we're going to have to craft our argument on those first two categories of evidence, strong epi data or associational relationships, and really clear understanding of where this sits on the causative pathway, which is fine. So I guess my second comment is related to how big a change is it going to need to be. So we've accepted, changes in GFR as well as as well as changes in proteinuria, actually, as surrogate endpoints, at least in certain disease settings. And I think, if you look at sort of these two buckets of endpoints, at least if you focus on GFR, if you think about the arguments supporting its use as a surrogate, you don't get to kidney failure unless you lose kidney function, right? And so whole lot of, scientific rationale for doing it. I think where we've gotten burned sometimes, though, is what we're really trying to understand is the effect on the irreversible loss of kidney function. And as you know from clinical practice and other things, you can have changes in GFR that don't reflect an irreversible loss. And so sometimes when groups focus on, even like a 30% decline, we've seen this in the CKD space, actually it doesn't always work so well as a surrogate, just because probably there's other factors that can cause variability in GFR. So just something to consider as you think about that magnitude of change in GFR, and it looks like you're going to maybe do further analyses. In terms of the bucket of proteinuria, generally, when we've thought about proteinuria as a surrogate, and again, when we're in a setting where we don't really have data from randomized controlled trials showing us that some small magnitude of change is going to predict the outcome, we sort of, rely very heavily on that scientific argument, why getting you that low, would make sense, right, that this is a good thing. So, for example, the complete remission you have, right, or complete response, that has a lot of plausibility, or shifting somewhere upwards from that. And we also very much anchor on the population having very high levels of proteinuria at baseline. So, I think, we're game for discussion of all of these things, but I think in a setting where you don't have data from randomized controlled trials, or bias until you get data from randomized controlled trials showing that your treatment effect on the surrogate reliably predicts the treatment effect on the outcome, we're going to want to see really big changes to think that this is really, and we can call it, because we like to categorize these as validated or, reasonably likely, et cetera. But if we're going to call it validated without data from RCTs, then I think we're going to want a really big magnitude of change.
Vaishali, did you want to ask a question, or if not, I'll ask a question?
But no, it just question, comment, whatever. So, when eGFR of greater than 25% was chosen by Laura Denver in 2005 at the Tours France meeting, it was arbitrary. We said 10%, somebody said 20%, somebody said 30%. And then we came up with 25% in 2005. And that has stayed along from 2005 to now 2024. So I think that we do need to kind of look at that, and I think that your study of testing cohort hopefully will tell us that what is the best ROC based on, thousands of patients that Dr. Mukhtar will collect. I do want to also ask formula for EGFR, because I know that the EGFR formulas are very different. CKD epi versus MDRD versus Cockcroft, I can't even say it, Cockcroft, whatever, right? Those numbers vary. And, it is little different. The renal staging that Dr. Palladini developed, the entire paper doesn't say what formula was used, but, speaking with him, it was CKD epi. So I think that will have to be, kept in mind. And I think the third thing I wanted to say was that the reduction in GFR based on just progression of AL amyloidosis is the surrogate versus variability, because they all have cardiac involvement as well. And we always talk about cardiorenal/hemodynamic effects. And, now, proteinuria is getting better, but, your EGFR is getting worse. And is this really progression of renal AL amyloidosis, or is this progression of something else? And the last point, and I'll stop talking, clinical trial. So, you are going to validate this in clinical trial setting, and we have a lot of AL amyloidosis clinical trial that you could validate it in, but you are not going to have really proteinuria every month on all these patients, even in clinical trial, because we are collecting them every 3 months. So I think that you will have EGFR or creatinine, but not really proteinuria measurements on a monthly basis, because, we generally have done it every 3 months.
Angela? Hello? Down at the end here.
Oh, right.
Yeah, I have been struggling with endpoints for 30 years, so I am extremely impressed and inspired by the forum this year. I have 3 points to make. The first is that 2018 Pavia paper, okay? It had nothing to do with progression. It had to do with time to next treatment. And the 20 DFLC was regarded as a high risk of DLC progression. When they did the multivariate analysis, it didn't fall out as significant. The only thing that fell out as significant was cardiac progression. Okay, so I do think it's important also to note that the time to next treatment was dependent upon the opinions of 2 amyloid physicians. The population, the study population, importantly, were people who had responded to initial therapy and then were in observation and progressed at about 3.5 years or so, and were deemed subjects for second-line therapy. That was the terminology that was used. So it does raise the issue of constructing clinical trials for certain study populations in AL, which I'll get to in a second. My second point is that the work done with organ responses, as Ellie and Vaishali have been working on, is extremely important, because we now know that there are drug developers who will target the organs. In the Kidney International Reports paper we just published, based on biopsy material from renal amyloid patients, the PI3 AKT pathway appears to be extremely important for the development of fibrosis. And there's rich data there that could be used in an experimental model to identify specific molecules that could be affected with drugs to stop renal progression. I don't think the potential exists to reverse renal damage in AL beyond a certain point. The same applies to the heart. We don't know enough about the microenvironment of the organs, and we don't know enough about what's happening to the fibrils, because the fibrils are being degraded. And if there's still pathologic light chain, new fibrils are forming. So it's active tissue. It's not to be thought of like the pictures we see of fibrils, where they're just little elements there randomly displayed. There's activity going on in the tissues. The final point has to do with study populations. There are basically three study populations. They're newly diagnosed. There are those who don't respond to treatment, newly diagnosed who don't respond, and that's about maybe 15% of those who get treated with Daricitabart-D, maybe a little more, maybe 20% of those who get treated with Daricitabart-D. And the relapse population. So there's a first relapse population, and then there are patients who get 2 and 3 lines of therapy. So your work trying to define progression for those different study populations is extremely important. And I would urge the companies who hold the data that you described to allow you to analyze it. I don't think you're going to be able to come up with a simple number. You know, I think it's going to be more complicated. And we have data safety monitoring boards for clinical trials, but we may need to expand that concept for AL trials. There may not only need to be a data safety monitoring board, there may need to be you know, an eligibility assessment board and a progression assessment board. So some kind of construct, given the complexity of AL, may need to be conceived of.
You make great points, Ray, but, you know, that last bit, I think it's challenging, right? You know, you need to draw the line. You need to draw a line somewhere, and nothing's ever going to be perfect. And maybe you draw a different line in a different population than in another population, but you need lines. No, I don't think any agency is going to say, and the committee said, you know, that this is a progression, therefore, you know, we have this many progressions, let's approve the next drug. So nothing's going to be perfect, but we need to understand, like, where the imperfections are, and, you know, by doing different sensitivity analyses or whatever, perhaps we can find, like, where that might be. And like you said, different populations, no question. You know, you say that 15% may be refractory to Dara-based therapy, but it depends. That refractory is in the eye of the beholder. You know, there's some people who say, oh, they didn't get, you know, a CR, therefore, you know, they need more therapy, and that's a different kind of, I mean, there are different people who say different things, and the way you view it. But I think in terms of, you know, the opportunity to understand the true biologic variability of these assays and then understanding them in various populations, I think is really going to be absolutely key to us. Yeah. Hey, Spencer.
Hey, how are you? I'll try to get closer this time. So just a request, I think, for the group. The work was really great here, and this is in relationship to the renal endpoints. And I realize this is sort of outside of the scope, but as you guys know, there are a lot of the other rare amyloid types who suffer from renal dysfunction, and they progress at varying rates. And those groups are kind of, because they're so rare, hard to study on their own. So I think a nice future project would be able to look at those sort of combined groups of patients who suffer from rare renal amyloid diseases. And there's just not a lot of information on progression rates, good endpoints, maybe tougher project, but
yeah,
patients in desperate need.
No, totally get it. You know, so my understanding of our assignment is sort of shovel-ready, you know, kind of things that are practical, feasible. There's so many questions to answer, and, you know, there's so much need. But with that, I see 2 hands. I don't know whose hand went up first, if it's Ashu or Justin. We'll get Ashu. He's on the left for me.
Okay. Thanks, Angela. Great presentations and very nice discussion so far. I wanted to touch on the point which you made, Angela, in your presentation about complete response and the validation of the assays at the low levels of light chains. I think that's a really important point. And I think on one of your slides, you had the different definitions of CR, and I suspect one of the aims may be to validate what sort of CR is really important. When we looked at mass spectrometry and used the ISA criteria, 30% of the patients were mass spec negative. But when we use the Andromeda criteria, 60% of the patients were mass spec negative, and clearly those two criteria are telling us something very different. And we did not see a survival difference when we used the Andromeda criteria compared to the ISA criteria. So clearly there's some kind of validation of criteria to be done. And as we are adopting CR more and more as an endpoint for clinical trials, I think we need to have a very clear definition of what CR actually means. And then the second point around the same is the measurement of light chains. The free light assay, which we use so widely, has, as you said, a significant inaccuracy, particularly at the very lower end of the range. And I wonder whether as a part of this forum, we could develop a validation strategy, but maybe even consider working towards a quantitative mass spec assay for these low-level light chains, which might actually allow us to measure them accurately.
So I'm going to just respond to that. As far as the mass spec, obviously you did the work with the binding site, now Thermo with the free light chain, and that's really powerful. And, you know, at Mayo we have mass spec, but we don't have a free light chain, so we can't quantify. So we can quantify for intact, but not for free. I don't know the availability of the, you know, mass spec that the binding site / Thermo has. So I think it's has amazing potential. The question is, how do people get access to it? And so if something's amazing and nobody can do it, it's less amazing all of a sudden. And so, yeah. Anyway.
Take the point. And I guess the plan is to get it validated and in an automated fashion, eventually similar to the Extent assay. So that will be coming in due course, but having it quantitative at the moment is just a qualitative yes or no assay, and working towards having some quantitation will help. But I think the CR question is the other point.
Yeah, and the other thing with free light chains, I mean, Siemens has an assay, and then Sabia now has an assay. So, you know, most of what we've talked about is, you know, with using the binding site reagent, because that's what was there first. But anyway, so who is next? Is it Frank or, oh, Frank was next. Okay, Frank Brideau, Dr. Brideau. Are you there? Yeah.
Yeah, maybe, can you hear me?
Yes.
I'm sorry, it's quite late here in France, so thank you for the very interesting conversation. I've got some questions for Dr. Mushtar, and in France, we are mostly relying on spot urine albumin over creatinine ratio for evaluating renal response. So do you think this might be considered? And we also rely on serum albumin level, because, you know, some renal responses can be different, and what we expect in many patients is the correction of nephrotic syndrome. So I would like to know your opinion on that. And regarding the kind of formula we should use to measure GFR or to calculate GFR, I guess that CKD equation is the most commonly used in the world. So these are just my basic remarks. Thank you.
So thank you, Dr. Brideau. So in response, I think that I didn't mention in the presentation, but we do intend to include the serum albumin. I think that will be nice to see correlation and see how it works. Although I do suspect that it may be influenced by other factors rather than proteinuria, so I don't know if it will be as accurate, but it's definitely easier to measure. So this is its attractiveness. As regard for the spot urine, I think that we probably should include that because 24 urine is a little bit difficult on patients. I don't know if we have as much data as 24. I don't know what centers do. We customize for 24, so it might be a little bit more sparse to make a robust conclusions from that. But that's definitely a point that we can take into those data that we intend to collect. Thank you.
Thank you.
Justin.
I'll be brief, but I think it was Spencer that brought up the the less common amyloid subtypes in kidney function progression. You know, and again, this is not a question, but more so a comment, and I'll be very brief because it's slightly out of scope for the topic at hand today. But, you know, along the southern border, we do see a fair amount of LECT2 amyloidosis that does seem to be more common than we would think. and it's probably undiagnosed. So just, I think food for thought that may be, again, I use the word may because we really don't know how many people have it, you know, an opportunity to understand the natural history of that disease.
One other thought about the newer novel therapies for AL. So daratumumab is not new anymore, but all the bispecific antibodies, they drop kappa light chain levels, they drop lambda light chain levels, and then, you know, if we are talking about DFLC as an entry criteria for any clinical trial, it is absolutely impossible to measure because they remain undetectable for a while, even if they're off trial. So I think that, you know, and even daratumumab also suppresses both kappa and lambda both, and that's why we had to revise the ISA criteria to include that if IFLC is greater than whatever, or UFLC is greater than or less than whatever. So I think that DFLC and ratios are difficult in this newer monoclonal antibody and bispecific era. And should we be instead looking at IFLC alone?
So I'll try responding and then see what other people want to say. But I mean, the reality is if they're both so suppressed, why would you want to put them on another plasma cell therapy trial per se? That's number one. And a less than to me is basically you're subtracting 0 from the number, so you essentially have IFLC. And the only advantage that DFLC has over IFLC is that if you do have a mild renal impairment, you are at least accounting for it, and you don't have to kind of be saying this, that, you know, if this, then that. So again, just trying to get to as much uniformity as possible without knowing you're not perfect. But, you know, but the IFLC is hard in that way because you get a little renal insufficiency, and all of a sudden, you know, you have an IFLC that's like, you know, 6 or 60, depending on, and so that's harder to gauge. So that would be my reaction to that. The one thing Ray, please comment.
Yeah, you know, I was the Lone Ranger there in Andromeda insisting that the criterion had to be changed. And I got some pushback, but eventually the data held up, as Vaishali said. And all of the immunotherapies in the case of the bispecifics and in the case of CAR-Ts do drive the light chain levels below detectable limits. Now, what that means and how much of a reset in the immune system occurs, we can infer to some degree from the autoimmune literature, which is very limited. But the autoimmune literature indicates that with CAR-T therapies for lupus and for myasthenia, the autoantibodies that cause the disease are undetectable in many patients, and the immune system is reset. There's reset of the adaptive immunity, if you will. Whether that happens in AL or not is unknown, and we're looking beyond the free light chain assay, as you both have suggested. But I think we need, as Angela suggested, a deeper understanding of the significance of changes in the free light chain assay data. The other point is you struggle with these low-level light chain cases. It reflects the diversity of the light chains in the disease. Some are more amyloidogenic than others. Some are more dependent on concentration. And therefore, the numbers, you talk about threshold, the numbers you come up with are going to be problematic by definition.
Thank you, Angela. I'm glad to be part of this working group. Thanks for the excellent work. I just have some couple of comments from industry perspective. As when we designed the clinical trial, you know, as Commenzo said, the first important is you cannot take out the context of the patient population, right? The endpoint is associated which population we studied. And also in the trial design, you have to decide what is the primary, what is secondary, or the exploratory. So all these endpoints are not weighed equally. So I think for working group moving forward, we need to talk about specificity, right? So what is relevant to which particular population? What is hierarchy order in terms of primary endpoint, FDA accepted, patient relevant clinical outcome, right? So, and I know there was composite endpoint is not new, right, in the trial design, but I think there is a thinking. So amyloidosis is multiple organ involved disease. So I want to ask for the regulator here, what is the likelihood in terms of feasibility of combine different organ at endpoint, for example, cardiac and renal, or other interesting combination? I mean, because when we, as a sponsor, speak to one of the division in FDA, we don't typically engage multiple divisions. So how that going to work moving forward? So this is a question we are having at the sponsor perspective. Yeah, maybe perhaps somebody from Regulate could comment on that.
And as Dr. Thompson's coming up, I'll just say, so we weren't dealing with all endpoints, unfortunately, in this, and sort of just we were told to pick a few things to present. So, but they're great points that you're making, and please do address.
Yeah, and one is just sort of a process issue. Like if you have some sort of endpoint, you're seeking claims that affect multiple organ systems. I mean, how we work as an agency is we usually say, okay, I'm going to pull neurology in if we're the primary division for something, but it has, more of a neurology endpoint, right? So we do talk. We do bring groups in. In terms of composite endpoints that sort of include multiple organ systems, I mean, conceptually we do this, meaning, for example, in our renal outcome studies, we combine, often the composite endpoints look at both CV death as well as sort of this marked loss of kidney function or progression to dialysis. That said, there's a complication. It's not straightforward exactly how to do this. And you could also imagine that even though you throw a bunch of things into your composite, the reality is you only affect one of these. So probably merits further discussion here. But regardless, whatever you throw in there has to be either a validated surrogate or a clinically meaningful endpoint.
Yeah, thank you. I have one more comment. So, Angela, you mentioned about variabilities of the endpoint, and I think another thing for us is important to address is missing data in clinical trial. I mean, this sounds like data collection issue, but I think it's a totality of endpoint. When you have event-driven study, you have a missing data, that affects the result, right, outcome. So I think from analytical perspective, that's a thing we have to address to regulator, how we address missing data, right, in terms of integrity and the totality of the clinical trial data. I don't know if we have answer, but obviously that's one of the components to be considered. Yeah.
I think that's
you're absolutely right. I've been on adjudication panels for important drugs where there are 3 doctors who adjudicate responses. And if 2 agree, the response is stamped. If 2 disagree, the third breaks the tie. And much of the data in an important phase 3 trial there were gaps, and you had to use inference in an individual case to gauge response. So you're absolutely right. It's extremely important. And having been chairman of data safety monitoring boards for large phase 3 international studies, it's a challenge to the sponsor to try and get the data complete and in a timely fashion. One final minor point is with respect to composite endpoints, and that is that major organ deterioration, PFS, was a composite endpoint. And I think refinement of that endpoint along the lines that Angela is suggesting may be particularly useful for the relapse population. As you could see from the data for progression to dialysis, the patients who are later in the course of amyloid do have a rate of progression to end-stage renal disease. And based on the PAVIA data from the 2018 paper, cardiac progression is also predictive of survival in the relapse population. So MOD PFS may be useful in that population with some tweaking of the criteria for progression.
Yeah, I agree. So again, the idea is because one of the elements of MOD PFS is hematologic progression, you know, in order to have a good composite, you need good components. And right now, one of our components is broken. And so the question is, you know, so not the end solution, because there's a lot of work that can be done, but, you know, just one of the really fundamental components. Let's understand it better. And it's both the free light chain changes and the fix. I mean, there's variability there too. So both of those aspects, and really just understanding the limitations and what those are, because then that informs us how we can put it into the next maybe MOD PFS or the next whatever composite is. But we're just so stuck if we can't use this, if we don't know how to use this. And so that's where sort of I was focusing. But every point, you know, the whole idea of confirmation. So not just confirmation of progression, but confirmation of response. You know, how many measures do you need before you call that, you know, your best response, and then you calculate from there. Like really formalizing what is that? You know, and then when you have it, you know, how do you deal with the multiple points? Do you average them? You know, or do you just say like the last one, the first one, like, you know, formalizing how you're dealing, how many measures do you need to have a best response, or it was just, you know, a random thing that it was really low, you know, and now we're going to measure off of there, and then we go up. And so, I mean, there's a lot of rules, and there's a lot of room for improvement, no question, but let's trying to get to like just one of the basic areas to go. And so we need everybody's help to do that. I'm going to ask a question about, again, the idea of, again, thinking about renal progression, because we all know, and Vaishali said it too, is that it's not pure. I mean, and Dr. Thompson, you were talking to that too, is, you know, it's temporary, you know, and it's not permanent, so how do you use that? And so when one is thinking about renal endpoints as an example, you know, how does one really deal with EGFR? Because, you know, there's there's toxicity, you know, they were septic, and so they had a hit, you know, so that's an attribution thing. Maybe we can understand that better. But, you know, they have reduced cardiac output now, and that's where, you know, the EGFR goes down. And maybe that's not a bad thing in terms of, you know, that it's one test serving many functions. But if somebody is using a drug that is, you know, supposed to help organs, so to speak, you know, how would that work? How would that be perceived? Or do you have to show, well, it's independent of, you know, cardiac function, or like, yeah.
Angela, one test that maybe can be done, sorry to interrupt, is to look at only cardiac patients with no renal involvement and see what is the EGFR change. I assume that it might have some survival impact, because losing kidney function by any mean is not good to any, you know, person, not just, you know, patients with AL. So that may be kind of maybe another way of elucidating a little bit the contribution of EGFR in general, decline in EGFR or change in EGFR to outcome of patients irrespective of their renal. Some people have renal involvement that is not glomerular, so they won't have proteinuria. So it may be a progression in the kidney, but we just don't know because we don't know that the kidney is involved. I mean, there are all kinds of ways that we can probably look into that.
Absolutely.
Yeah, and again, just to speak to this issue, you know, because I think where we did a lot of the work with GFR and proteinuria, or at least GFR, was sort of CKD progression, right? And figured out various ways to define it. You know, started with the doubling and then got down to 40, and in some settings looks like you could use a 30, and in some settings you can use a slope, right? But if you take that paradigm and everything we learned there and try to plop it into other disease settings, not clear that it works, right? Again, gets to this concept of you got to validate your surrogate endpoint in your population. I think ways in which we protect ourselves, So, for example, with the GFR issue, my sense is if it's a large enough decrement in GFR and and you show that it is sort of sustained, that you are going to protect yourself, right? So again, if you focus on the 20, you may find yourself running into issues here. But if you really have a very large decrement, you're probably going to be helped. I think you're also helped, to the extent that you're not just looking at one endpoint, but you're looking at, if you're talking about, again, AL amyloidosis and they've got a ton of proteinuria, you're looking at GFR and you're looking at, proteinuria, right? And so you got a package of data. But even when you sort of, have those two considerations or even consider that you think you have some definition that you think is sort of going to be your clinically meaningful response that everyone can say, this is it, it's validated, we all feel good. Anytime you put it into a drug development program, you got to think about the individual drug, what you know about, the pharmacodynamic effects, what you know about potential off-target effects. So I think, even if something is a validated surrogate endpoint, doesn't mean that in every single drug development program it can be used.
Yeah, I think I view this discussion a little more positively, I think, than everyone. I think we have 3 completed phase 3 trials in TTR, some coming. I have to say that the patient's eGFR, at least in our data set, goes down by about 8% per year in natural history studies. And my sense is taking care of these folks, when they develop advanced disease, they go into a cardiorenal syndrome, and their cardiac output goes down, their JVP goes up, and their renal function gets worse. So I wonder, maybe Eli was getting to this, we have the opportunity right now to probably pool all the data. And if the gold standard is data from, multiple randomized clinical trials in which different mechanisms were applied, we may have available data to validate if eGFR is something that can be used, at least in TTR amyloid, which I think could ultimately inform AL as well, but just hypothesis.
I think I want one correction. It doesn't matter where you got the data from, whether you got it from an observational study or a registry or an RCT. The whole issue with that sort of highest level of evidence is that you have to show that treatment effects on the surrogate reliably predicted the treatment effect on outcome. And to the extent that you're validating the change in GFR to the change in kidney failure, your treatment would therefore have to be effective on the outcome of kidney failure, right? And so I think that's the nuance here, which you guys aren't going to get to. So what we're probably going to try to get to is something that you can call validated based on sort of just such tremendously strong scientific rationale, observational data, et cetera. But could be wrong.
To Matt's point, Julian's going to present data on looking at renal function in ATTR later.
I guess the question is, what if a decline in eGFR in all these clinical trials actually was associated with the outcome of survival, ultimately? Would that be?
So I'm going to tell you that we have probably, that just because something, I think the question fundamentally is, what is my surrogate a surrogate for? So I'm going to assert that if you're trying to validate GFR as a surrogate for something, that you should be trying to validate it as a surrogate for outcome, because you're never going to convince me, at least, that if you tell me what the treatment effect on GFR is, that I know what the treatment effect on mortality is, right? Like, that gets really dicey, because there's a whole lot of different ways you can affect mortality. And so, I understand, and I've seen this a lot of times where people sort of say, oh, and this is how, changes in GFR relate to your mortality. But the reality is what we're trying to understand is to what extent the treatment effect on GFR predicts the treatment effect of kidney failure. And you're nodding your head, so yeah.
So in terms of, so there are therapies that are directed at the amyloid specifically, right? And so more and more targeted therapies there. And so I don't think mortality would necessarily be the, I'm talking to Dr. Thompson and she's talking to Dr. Maurer. So, no, I was just going to try to address sort of what you were saying a little bit, because there are trials that are targeting the amyloid specifically. I don't know that mortality is necessarily the endpoint. So that's not something that we were talking about here today. But certainly understanding, I have this drug in AL that does nothing to the clone, but is actually supposed to help remove the amyloid from the tissues. And looky here, the kidney, is better or whatever. That is important. And so, separate from survival, but literally, so in that, I think there is a lot of import. No? Yeah, I mean, so it's not always all about mortality. So I think I heard you, but as long as we show kidney.
Well, I just want to say that when I think about, having been in a dialysis and been with patients, right, who are on dialysis, and I think about myself, when I think about an outcome of having to go onto dialysis, that's probably, some people might say that's worse than death. So you don't need to convince me that something that's a surrogate for kidney failure is.
It was a question, not a comment. It wasn't trying to teach you something. It was actually asking you something, in all truth.
Yeah, but hopefully I answered it.
Okay.
Yeah,
thank you. All right, we have a question here from, Julian.
Yeah, thank you for a fascinating discussion. I just wanted to respond to something that Dr. Thompson mentioned earlier, and that is the need to combine, from a renal perspective, combine GFR with proteinuria. I think those of us who treat AL amyloidosis have, come across patients who start off with a GFR of 90 and heavy proteinuria, achieve a complete response with their chemotherapy, and wind up with a GFR of 55 and no proteinuria at all in a complete response. And in our experience, so they would be classified as a complete responder hematologically, but a renal progressor. And in our experience, if those patients stay in a complete clonal response, they very, very rarely and almost never progress to end-stage kidney disease, which is ultimately what affects their life and morbidity. Those patients who've no longer got any proteinuria, if they haven't got cardiac involvement, they feel completely well. And so I think, it comes back to what Dr. Thompson was saying about the need to really combine the biomarkers that we do have, even with established biomarkers such as renal function, to come up with combinations, if you like, that make the best clinical sense, which at the moment I think they probably don't necessarily.
So there's a question from Dr. Buxbaum on the line. He texted in about cystatin C instead of, creatinine GFR, but cystatin C and cystatin C GFR. Any thoughts about that and the role for that, especially in elderly who might be, have less tissue, muscle mass, and so on?
Yeah, and I think this is a critical issue. I believe, if not you guys, at least others who are members of this group have thought a lot about this and are trying to do additional work on this. Yeah, no, our estimating equations in these populations, they're probably not the best out there. That said, so it certainly merits further discussion as you think about this sort of, this issue. But the reality is probably in your trials, all you're going to have is a serum creatinine or maybe a cystatin C, so you're going to have to work with whatever you have. But do very much recognize that there are these issues with the estimating equations.
Should that be put into trials, to learn moving forward, the cystatin C? Or?
Yeah, no, well, certainly, to the extent that in your associational analyses you have data for both cystatin C and creatinine, you could potentially look at this issue. But, yeah, no, I don't know the answer, but I think it's a very valid concern.
I have a question for, we have such a distinguished group here out in the audience and by phone, and I know at least in our group in PRO endpoints, we had 7 ideas, and that was just in an hour, I think, that we came up with when we were meeting. And we kind of tried to narrow that down, and the top 3 ideas were, providing the evidence or doing studies to understand the evidence around using KCCQ, as we talked about. There was the systematic literature review and ATTR around the PRO measures, and there was potentially development of a AL measure, the one that was presented used items from existing measures, but was not developed out of and by patients. So there was that idea, and then there were several others. But what other things do you guys have? Do you think that our ideas hit the mark? Do you think, hey, we have a limited amount of resources? You know, I'm just really curious of what we might have missed or what ideas that the group has.
And even though CPET wasn't in this session, they did talk about it as an endpoint. So, you know, you can include things that were discussed elsewhere or other time. Please, I think that's great. We'd love to hear thoughts. Nada.
Everybody's ready for break.
Well, we certainly can break early. Got great.
Hi, Michelle Watson, Novo Nordisk. I was actually wondering perhaps on the PRO journey, if you considered in the work with KCCQ, deciphering health state utilities as perhaps a future project, because that might be valuable in terms of being able to quantify the actual outcomes of interest.
That is a really great question. That is not something that we did so far because at the time we were looking just for, you know, efficacy endpoint rather than health state utility and considering that approach to it. So that is one thing that I think we should add, maybe not specific to KCCQ, but overall looking at health state utilities and how they are used. We know there are a lot of potential downsides of some of the current measures that are used that include health state utilities, and further examination of those and some recommendations about what would be appropriate. Like we know there are a lot of generic tools, for example, EQ5D, et cetera. We know there's ceiling floor effects. What could we do to do a better job of estimating health state utilities? Is a disease-specific measure for health state utilities required, which would not be KCCQ. That's not what it was designed for. I think that's a really good point. I think we should write that down and consider that. Thank you.
I'm going to let Julian go first because he had his hand up first, and then Sharmila.
Thank you, Angela. A question along the same lines, really, in that, you know, I wonder whether the PRO is a chance really to get away from the fact that certainly in ATTR amyloidosis, we've needed polyneuropathy studies and cardiomyopathy studies as separate studies. And, you know, it's meant doing with the same drugs, 2 different trials at 2 different costs, 2 different times. And so far, every drug that's benefited polyneuropathy has benefited cardiomyopathy and vice versa, I think it's fair to say. And I just wonder whether a PRO is really a way, or defining a PRO that deals with our multi-system diseases is really a way to try and get away from that labor-intensive model that we currently have of looking at one organ and different organ and in different studies.
I think that's a really good point, and one that we hope to find more out about. Back in the 2018 meeting, we had come to the conclusion that an ATTR-specific patient-reported outcome measure was needed, and that's now been developed, and most all of the validation efforts are completed. We're now waiting to look at responsiveness to treatment and meaningful within-person change, and are basically awaiting its use in studies that would have that data to be able to look at it. But that particular measure, the ATTR-QOL, was designed with that multisystemic sort of all-encompassing view for both symptoms and impacts, with multiple different scores that could come out of it. And there is a little more work to be done to understand how that could be used as an endpoint in these different trials. But I think we are so far along in that really is a good point. I welcome comments from others on that. I think it's missing in AL, though.
Thank you so much. I think I'm just continuing on Julian's point and others' point. So we recently looked at our AL cohort and found that the functional measures. In our case, it was Minnesota Living with Heart Failure. Vaishali is aware, as part of the study, 6-minute walk test. And what we found was that they were independently prognostic over and above the imaging markers. So my question is, did your group consider combining the PROs with some kind of imaging markers to enhance some specificity or maybe actually enhance prognostic utility in combination?
Our group specifically has not looked at it as a composite measure because there are so many complex issues just involved in a PRO score to begin with. There was a group with the 2021 that was looking at composite endpoints that was separate from this. So I would welcome FDA comments on that, but I do think it would be hard when you, with the PRO endpoints, have a multi-item score with a single domain or could be multiple domains under it, and then you also would have to meet a threshold on some other markers. It might be better to think about your endpoint hierarchy and having it separate, having maybe separate exploratory endpoints that include some of these and see how that comes out first. I see some nodding, some of you, so I welcome those comments.
Yeah, I think it's going to be challenging to be able to interpret a composite of a PRO and maybe some kind of an imaging biomarker, which is not a validated surrogate endpoint. If you wanted to look at it from the perspective of maybe a prognostic marker, go for it. We're not going to ask much depending on, whatever your data shows in terms of if the prognostic value is enhanced, if you wanted to use it as an enrichment factor. But speaking of composites that have maybe different levels of clinical relevance, there are ways to evaluate those types of composites and maybe analytical ways to go about it. But I think interpretation is very challenging. So I just wanted to throw it out there. Evaluating as primary or secondary or exploratory endpoints just to support the primary findings is maybe another way. But yeah, definitely a biomarker that's not validated, I think can be, one, not acceptable, second, just the interpretation challenges.
Sorry, just a quick comment about, this is not a new endpoint, but perhaps the analytical methods by which we analyze these endpoints. We all know about the hierarchical methods that were used in the cardiomyopathy trials with ATTR. I think that should be applied also for AL, and how many endpoints are included in that hierarchical analysis to be able to, as efficiently as possible, develop new drugs. So that might be another topic for your group.
Thank you.
Yeah, just a quick follow-up to that comment. We try to discourage combining. You have various hierarchical methods to analyze, as example, win ratio. But when you are combining mortality, heart failure, hospitalization, and then 6-minute walk, KCCQ, or maybe throw in a biomarker, I think the interpretation becomes very challenging. If you've outrightly won on the more important clinical endpoints, we can work with it, but if you have small changes on all of these endpoints, we're really struggling to interpret what is the real treatment effect. So it's not just about getting a statistically significant result, it's also about how are you getting there and what is the real treatment effect.
Any more takers? We want to hear your thoughts. Or we could end 1 minute and 50 seconds early for break. I think that the votes are in. Okay, thank you all. Bye-bye.
Hi, everyone. We're going to get ready and go ahead and get started. This will be a largely virtual presentation, but we'll have several folks joining us here in the room from industry, as well as our regulatory colleagues that are here. And so this is a session that will focus on the Federated Analytics Working Group, which is being chaired by Dr. James Signorovich of Analysis Group. I am delighted to turn this over first to Dr. Preston Dunman, former FDA Cardio-Renal Division, Cardio-Nephrology Division, as well as former Janssen R&D. And so I'm sorry, Dr. Dunman, I'm trying to advance to your slides, but they'll come up in a second, but I'll turn it over to you. Thank you.
Sure. Thanks so much, Kristen, and sorry for the scratchy voice today. We've heard much today about the expanding number of data sets that exist in the ATTR and the AL spaces that include numerous clinical trials, institutional clinical data, and potentially biobank laboratory samples and even images. The potential use and pooling of these various data sources hold great promise. However, as we all know, this is not always a simple matter, given the need to safeguard the proprietary nature of these data sources, as well as to remain in compliance with international data privacy rules and regulations. One avenue to address these concerns is the use of decentralized, federated analytical data systems in which the patient-level data remains behind proprietary and legal privacy firewalls of the data owners. Many of you know that ARC has been working with Dr. James Signorovich to explore federated data analysis for quite some time now. Dr. Signorovich is a statistician and managing partner of the Analysis Group in Boston, and he's kindly agreed to be the subcommittee chair of our Federated Analytics Working Group. So I'll turn this over to him now for an overview of that work, followed by a proposal from Dr. Gillian Gilmore from the University College London for a federated analysis project regarding the decline in renal function in ATTR cardiomyopathy patients. So James, take it away.
Great. Thank you, Preston, for that introduction, and thanks to ARC for making space for this discussion. I think there's been many references to federated analytics throughout the day, and so we wanted to take some time to talk through, what this is, what are we already doing, and what's the progress on that front, and what does this require going forward. So I think the key thing to keep in mind for federated analytics is the way we're thinking about it within the forum is really shared learning without waiting for shared data. We're going to go into a lot more detail about how to do this, but this is really the role it plays when we think about doing collaborative research within this group. And I think the forum is such an excellent place, as we've been hearing, to identify and refine research questions that are important for patient care and for drug development. And I think what we'd all like to do is be able to answer those questions in as timely and high quality a way as possible, drawing on, in many cases, data that might already exist. But as Preston nicely laid out, that's not always easy. Even if data exists, there are often real and understandable hurdles to data sharing or timely data sharing across institutions or across international boundaries. So what we've been doing within the Federated Working Group of the Forum is looking at the different research goals that are prioritized by all of the working groups and trying to help identify those that would benefit from federated analytics and where federated analytics could be feasible, and then help the research teams explore that path forward. As mentioned, we within the forum, we have already been advancing analytics in a federated fashion, and I'll give an update on that, that research. So the overarching goal that we took on was to assess the evidence for surrogacy of NT-proBNP for survival in AL amyloidosis. And the gold standard evidence for doing this is to look across multiple randomized trials look at how effects on NT-proBNP correlate with effects on survival. And when we started this research, the only randomized trials out there were coming from 4 different sources. So that's the Andromeda trial from Janssen. There's also the Tourmaline trial, which we heard a bit about earlier today from Takeda. And then 2 other trials. One was a European Hematology Group trial, EMN03, that was housed at Pavia in Italy. And then a trial that was conducted at Nanjing University Medical Center. The data for that was housed in China. So I think we can all appreciate that the chances of pooling all of these data from these different sources in the same place anytime soon is very, very low. But to do the research that we prioritize to understand the evidence for or against surrogacy, it was really important to look at all of these data and to do so in a harmonized way. It was not possible to just use what was in publications. We need to make sure NT-proBNP response is being defined consistently, that we're looking at the response evaluable population for survival. So all those details that are critical for doing this in a high quality way would really require doing new analyses of the data in a harmonized fashion. So the output of this, which we presented at IAS, interim output, is shown on the left. And what we've done so far is really replicate the known association between NT-proBNP response and survival. This is not a new result. This has been seen from a number of registries. But we took this as both kind of practical and scientific validation of this federated platform approach, where we could replicate this known result by looking across multiple trials and aggregating the findings. Our next step is to generate evidence such as that shown on the right. This is just an example taken from solid tumors, but this is that kind of gold standard analysis where we're really looking across trials at the association between effects on the possible surrogate NT-proBNP versus effects on survival. So we are enormously grateful to Janssen and Takeda and to the group at Pavia and the group at Nanjing for their contribution to this study, both in allowing their data to contribute and also the work that they put in to collaborate with us and help run some of the analyses that we provided on their data. So the current status is we have an analysis plan that was developed within the forum with many collaborators, including from FDA and MHRA. As I mentioned, we've replicated those predictive associations. So the next step that we've been waiting for, and which is we understand is coming soon, is to really incorporate additional mature follow-up data from Andromeda, and then to conduct those surrogacy analyses according to the SAP. We've also, with the collaborators, expanded this to look at not just NT-proBNP response, but also NT-proBNP progression as a candidate surrogate. So our next steps are really to update the data, run those analyses as planned, and then move towards publication of whatever it is that we find. As we've been conducting this, there has been a lot of interest from many of the collaborators in looking at the possible surrogacy of different measures. I think hematological response is high on the list. We were just talking about renal response in ATTR, but that may be of interest in AL as well. And also composite measures. It is a multi-system disease, and maybe the best surrogate is actually a composite of many biomarkers. So we wanted to say a bit more about where federated analytics fits in kind of an overall research strategy. It's not something that we would always use, and it's also not a silver bullet, but it has an important place. And so the way we think about it is if as we're evaluating research goals and looking for what data exists and whether it's adequate for addressing these goals, we start to see where does that data lie, right? Does one center have enough to do the research on their own? Does someone else have it out there who's willing to share the data? If we're on that path where the group that is capable of doing the research and has the resources and expertise either has direct access to the data or the data can be shared with them, then we don't need federated analytics. Things can proceed in a traditional manner, data sharing agreements, however it can get done. But as we all know, that's not always possible. If the data are sparse or distributed across multiple centers, trying to get data sharing agreement in place for everything could be time limited and time consuming, and that might be a major hurdle. It might be that even just to get a feasible sample size, we need to look across multiple centers, or like in the surrogacy case, like we really need to look at multiple randomized trials and find a way of doing that when direct sharing is not possible. So it's really those situations where direct access to patient level data, can't happen quickly enough, where we would look to federated analytics as a possible solution. And so to give a little flavor of like how federated analytics works, how do we do the analyses when we don't have direct access to the data? The key approach is we are harmonizing the data that sits locally at all of the participating centers or institutions. So all of the centers have some type of database for storing their data. And what we need to do is just take the data that we need for our research, put it into a common format, do some pressure testing, understand the ins and outs and nuances of each data source. But that's really the big lift in the beginning that, again, we went through, with Pavia and Nanjing and with Janssen as collaborators. Once the data is organized in that fashion, a group can develop, do the analysis programming centrally, and then distribute it to all the centers where they can run it locally, get summary results like, tables or figures, get those readouts locally, share them, and aggregate them all into the final analysis. So no patient level data needs to cross institutional boundaries, and each group, has control over what is getting shared and aggregated. As we develop, as research questions arise, and we sort of start going down that path of seeing whether a federated approach is needed and is feasible. An important question is, is the type of research or the type of analyses that we want to do feasible with federated analytics? Most things are feasible, and the real question is, how hard is it? How much time and resources would it take to do this in a federated fashion, and what are the strengths and limitations of different approaches? Generally, anything that looks like a meta-analysis is going to be on the pretty straightforward side to do with federated analytics. But a lot of things like descriptive analyses, correlations, regressions, and many of the kind of traditional statistical analyses we want to do are quite feasible with federated analytics. When things get more advanced, certain types of machine learning or like patient one-to-one matching approaches or more complex analyses, we need to proceed carefully. Those might be feasible, but it might be the more work than necessary to make them feasible through a federated approach. It's always best to evaluate the feasibility, both technical and scientific, on a case-by-case basis. But one thing I think it's important to emphasize is part of the value of federated analytics is that we don't need to have adequate sample size at each and every center. So even if we're interested in a very rare genetic subtype where no individual center could do an analysis that would give sensible results or be worthy of publication, that might be a case where we really need to look across centers, and the federated approach would have high value for making sense of the kind of the larger sample size of relevant data, even though it's sparse and spread across multiple institutions. And of course, it's always important to be very careful about harmonizing the study measures and definitions, and if it's not possible to harmonize them, to be aware of where the differences are and how that might impact the study results and interpretations. So to facilitate the feasibility assessment for different research goals, be they federated or otherwise, one of the things we've done within the forum is develop a database of all of the randomized trials in all forms of amyloidosis. So this was based on a systematic review, both of publications and then reconciling that with clinicaltrials.gov and extracting all the information available on who's in the study population, sample sizes, and importantly, the different study measures that were applied in the trial. Not just the primary and secondary outcomes, but any of the other measures that we could find in the public domain. So our hope is that this can, as research questions come up and we start thinking, hey, maybe we can look across clinical trials to help answer this. This would help us, hone in on first whether that's feasible at all, and secondly, which clinical trials might have adequate information. Of course, if the study, some of the study goals might be feasible through a meta-analysis of published data from these trials, that's great. Others might need to access patient-level data, and that would require either data sharing agreements or a federated approach. So I wanted to take a moment and highlight what we want data holders who might be interested in federated analytics to understand about the process and what it takes to participate. So the first key message, you don't have to share patient level data. It's going to stay locally as you currently have it. Second key message is that you maintain full control of how your data is used. So participating in a federated platform, for one initial study doesn't commit you to using your data for future studies. You'll be able to make that choice study goal by study goal through whatever internal processes you have for deciding whether and how to participate. Third, there is analytical effort that's required at each center, but it's less than doing a full study. So there'll be some effort to harmonize the data. That is something we provide support on, and can meet with the team and talk over Zoom and make sure we're getting the data set up efficiently and we all understand the ins and outs of it. But once that's done, your research team would receive analyses codes that are developed centrally and just need to run those analyses and then share the results. So it becomes a much lower draw on everyone's limited analytical capacity. And then finally, I think prospective collaborators should view this as an opportunity to contribute their data to important research that the community has prioritized, but also as an opportunity to advance research goals that you might be prioritizing. So you might have things you'd wish to learn and would really benefit from learning from data held by others. And so by participating in this federated approach, I think everyone would be good citizens, and there's opportunities to advance research that might otherwise be challenging to get answers to among your priorities. So we've heard a lot about federated analytics today. I think the list of study goals that we need to evaluate is probably growing. But just to recap, there are 4 that we've evaluated in depth. We've talked about NT-proBNP and other surrogates. There was discussion earlier today about CPET, and I think it's an open question of whether we need federated analytics there, but that's something we've been looking at. But we wanted to highlight a research goal that Dr. Gilmore proposed for looking at renal biomarkers. as one that we think is an excellent candidate for a federated approach in ATTR-CM. So with that, I'll turn it over to Dr. Gilmore to talk through this research goal, and then I think we're going to come back to a facilitated discussion.
Thank you very much, James. Are you happy to? Yeah. So thank you. I'm sorry not to be there in person. It's been an amazing day, but I'm going to present, as James mentioned, an idea for one potential project that may be amenable to a federated analytics approach, really in order to better understand the clinical relevance of decline in kidney function, which is so common in patients with ATTR amyloidosis as the condition progresses. James will then give us the clever stuff on how we might achieve it, as it were. So the overall aim really is to develop a definition of deterioration in kidney function in ATTR amyloid cardiomyopathy at 12 months from diagnosis that's got prognostic significance, and evaluate both its suitability for use in clinical practice to perhaps determine failure of response to treatment, but also in clinical trials as a potential surrogate endpoint, either alone or as part of a composite. Next slide, please, James. So why is the project needed? Well, a degree, as many of you will be aware, a degree of CKD is extremely common in patients with ATTR amyloid cardiomyopathy, and a decline in kidney function is very common as the disease progresses. A small proportion of patients do progress to end-stage kidney disease, which obviously does have a marked effect on morbidity. What about the supporting evidence? Well, the supporting evidence is very limited at present, but I will show you in the next slide some retrospective analysis indicating an association between renal decline and patient survival in ATTR amyloid cardiomyopathy. And I think the benefit for patients really is that it may indicate renal decline as an early indicator of disease progression, which has prognostic relevance, and it may thereby enable early assessment of, for example, failure of response to treatment, which is obviously going to become more and more relevant as we're likely to have several therapeutic options in ATTR amyloid cardiomyopathy. So this is really the supporting evidence that we have, which, as I say, is limited. On the left is the summary data from a retrospective natural history study of just over 2,000 patients with ATTR amyloid cardiomyopathy that we conducted. None of these patients, is important to say, were receiving disease-modifying therapy, and we defined renal progression as a 20% decrease in GFR at 12 months from diagnosis, and that occurred in 24% of patients. I should say, in response to Dr. Thompson's earlier comments, we did look at greater than 30% decline in GFR at 12 months, and we looked at greater than 40% decline in eGFR at 12 months. But in fact, the proportion of patients that achieved those cut points, if you like, was extremely small. And given that this is a population who are not on treatment, the feeling is it would only get smaller if we were using it certainly as a clinical marker. But either way, renal progression was independently associated with patient mortality and had a hazard ratio for mortality among progressors of 1.7 compared to those without renal progression. The panel in the middle shows the relevance of NT-proBNP progression, defined again at 12 months from diagnosis as a rise of greater than 700 nanogram per liter and a greater than 30% increase from baseline, which has previously been shown to be prognostic. And when we combine that with renal progression, you can see that the risk of mortality increases progressively from patients who have neither marker of progression or one or the other marker, the middle line in yellow there, or both markers of disease progression. And on the right, in a different retrospective study, which included a smaller number of patients, I think you heard about this morning from Joban Vaishnov. You can see that albuminuria progression defined at 12 months from diagnosis as a 30% increase from baseline in urine albumin creatinine ratio was also associated with patient mortality. Next slide, please, James. So the research objectives are really to establish a definition of deterioration in kidney function in ATTR amyloid cardiomyopathy with prognostic relevance. As I say, you've seen the data with loss of greater than 20% of eGFR, and evaluate the suitability as an endpoint for drug evaluation in ATTR amyloid cardiomyopathy, considering obviously event rates by baseline CKD stage, cardiac stage, other comorbidities such as diabetes, hypertension, gender, age. Obviously, and we talked about this earlier, the stability of progression or indeed response status over time is important. The associations between a cardiac response or progression and a renal response or progression, and the associations with change in functional status, i.e. 6-minute walk test or quality of life by KCCQ. And of course, the prognostic associations with longer-term changes in CKD staging, time to renal deterioration, and indeed time to end-stage kidney disease or transplantation or indeed death. Next slide, please. So what data do we actually need to address these research objectives? Well, obviously the target study population will include patients from the now multiple ATTR amyloid phase 3 clinical trials. And the minimum or key data elements that we think are required for analysis are at baseline are age, gender, race, diabetes, hypertension, obviously CKD stage and cardiac stage. And then at 12 months, we really need eGFR, albumin creatinine ratio, NT-proBNP, and obviously it would be nice to have functional markers such as walk test and KCCQ. And in terms of longer term data, we want more long-term data on kidney function, eGFR, and obviously mortality, both all-cause mortality and cardiovascular related mortality. And the possible data sources are obviously the completed clinical trials in ATTR amyloid cardiomyopathy subject to data sharing or participation in the federated approach. Obviously, the preferred group would really be pooled data from all arms of those studies. But the fallback option would be the placebo arm, the data from the placebo arm only. So it may be less suitable, having said that, for showing a wider range of changes and associations. So ideally, one would have the pooled data from both arms. So I'll hand over to James to tell us how this can be achieved. Thanks.
Yeah, and thank you, Dr. Gilmore. Collaboratively is the only way to achieve this. And the way we've envisioned this as, within the forum, there's such value from bringing together the patient perspective, clinical experts, regulators, and drug developers. So we're envisioning, everyone interested participating in the study design. ARC and their research partner would kind of do the research management and analysis implementation. An important role here for drug developers who are interested would be participating, either sharing their data, if possible, or if not, going down that path of participating in a federated approach so that they and the community can learn from clinical trial data in a timely way to answer these questions. And of course, this would be something that we would all want to publish to benefit the community. So I will close there and turn it back to Dr. Dunman.
I think you're on mute, Dr. Dunman.
Thank you. Thank you, Julie, and thank you, James, very much for very interesting proposal. And certainly we would be interested in the feedback of the community here. So while people are teeing up their questions, I might go ahead and ask a few questions of the discussant panel. Certainly, Janssen has been very active in sharing in this sort of federated experience with Andromeda. And so, Dr. Tran, thank you for joining us. If you wouldn't mind, share what your experience has been with that federated approach from the Janssen experience. Perfect.
So, but thank you for allowing me to participate here. So I'm Nam-Phong Tran, one of the medical oncologists at Janssen, who are leading the AL amyloidosis project at Janssen. So as you know, Janssen has already committed to contribute to the advancement of the AL amyloidosis. And when we were approached by James about this project, we do realize that we hold very valuable data from our randomized trial, of the phase 3 Andromeda study. And the process basically for us is actually quite simple, right? As James has explained, we were provided with sort of a programming code or analysis code, along with some data specification that we can set up and use those code on those data, and then provide the result back to James. So that part was simple. I think the harder part for us at that time was mainly the study was still ongoing. And there was certain analysis, like especially related to survival, for example, that we have not done a formal analysis based on sort of the timeline of the trial. And so we were not able to sort of provide that type of analysis, to James. And we weren't able to do the sort of analysis based on treatment arms because, again, the study was ongoing, right? So that's sort of the nuance. I think when we do this analysis is that, when the study is fully completed, I think it will be much simpler, and those nuance won't be there. But when the study is still ongoing, like our study at that time, it will take a little bit more sort of back and forth discussion in terms of, what type of analysis we can provide, versus what we are not allowed to touch in terms of the data. So that's sort of what our experience. I think this is a very important way of analyzing the data, right? Because first of all, as Jane has said, you don't have to basically move your data, so you have that sort of security to your own database. And the other thing I can see is that the cost efficiency, right? Because you don't have to move the data. An analysis, a central analysis code is being worked out and provided, and so it's actually very quick from Janssen's standpoint. Once we receive all that information, the turnaround time was within a few days, as long as you have internal resources to basically, apply that code to the data specification that were provided to us.
Wonderful. Thank you so much. Really appreciate that, Dr. Tran. I think we also have one of our discussants there in person, Dr. Alexander. Would you like to comment?
Yeah, so thank you for the overview of the federated data analytics. I think it's a potential powerful tool for the project outlined by Dr. Gilmore. The space is rapidly evolving in terms of our understanding of patient presentation, natural history, and treatment response. And I think we have to be nimble with the types of data sources that we use to keep up with contemporary trial design. And so completely agree with everything that was presented here. A little bit off topic of what we've shown so far, I think that this approach could also be very impactful for doing multi-center studies from academic centers, because these are variables that in our individual programs we can collect pretty easily. And if there's a way to harmonize it on the back end, I think that that'll be another kind of complementary approach to using trial data.
Excellent. And then from what I understand, Dr. Thompson is still with us. Dr. Thompson, would you care to comment from a regulatory point of view?
Hey, hopefully you can hear me. It's wonderful to see you, Preston. Preston used to work with us, which I think everyone knows, and then he left us. Yeah, just maybe a few quick comments. One, I just want to congratulate you. This is incredibly impressive. You know, obviously, patient-level data sharing can certainly transform drug development in terms of, you know, really advancing drug development by allowing us to answer really important questions. So regardless of whether all the data are all in one place or it's more a federated system, this is just really really wonderful that you have come together, and to those who have sort of agreed to participate in this, I would just say, wow, impressive. I think one of the hardest things that, you know, to have happen often is to get industry to share data. So this is just, you know, a tremendous resource for the community. I just did want to, though maybe comment a little bit on sort of the issue of surrogacy and the work that you're doing as relates to that. So, you know, obviously there is a toolkit that you need to manage patients, right? And you know, to figure out who has disease, who's going to progress, whether they've likely responded to treatment, to really tailor therapy to patients. You know, a separate concept, obviously, is the toolkit you need to advance drug development, and that includes the ability, obviously, to have biomarkers you can use or variables you can use for prognostic enrichment. Obviously, you need to have something that, you know, if for phase 2 studies, you know, response biomarkers, and then obviously there's the surrogate endpoint issue. I struggle a little bit with whether this work would help with giving you a renal endpoint that could be used as a surrogate, and I think some of this relates back to the discussion we had before. I mean, typically when we're talking about changes in GFR, you know, or even something like UACR, proteinuria as a surrogate, we're talking about it as a surrogate for progression to kidney failure, right? And I think, as noted earlier, just because you know what a treatment's effect is on, you know, GFR, I don't think it tells me anything about necessarily whether the treatment's going to have good or bad effects on mortality, or, you know, I don't think it provides any, you know, I'm not viewing it as a surrogate for a 6-minute walk or something like that, a cardiac function. And so I struggled a little bit in terms of sort of what you were doing here, and also got the sense as well, based maybe, I can't remember, Dr. Gilmore, if it was your comments, but I think the other fundamental question is this in fact a population at high lifetime risk of progressing to kidney failure? And I think part of this gets at the fact that at least, you know, at least chronic kidney disease is largely an asymptomatic disease, or if there's symptoms, they're nonspecific until very late. So this is not to negate the importance of the work that you're doing, potentially, you know, for various uses, but to struggle a little bit with whether, you know, this would get you to a surrogacy, at least the concept of using GFR as a surrogate for something, right? Certainly not progression to kidney failure, and would be wary of accepting as a surrogate for anything else, just based on sort of scientific principles, for lack of a better term.
Let's see. There's one other person here I was wanting to get to. Julian, do you have any comments that you want to make in response to what you just heard from FDA?
Just a question, really, I suppose, and that's the fact that, to my mind, one can, you know, we see a decline in GFR extraordinarily common in patients with ATTR cardiomyopathy, and to my mind, if patients, you know, if one diureses a patient to try and improve their fluid balance, their GFR is quite likely to decline. So to my mind, the combination, if you like, is the combination of using that along with NT-proBNP is actually quite attractive, because, you know, if you squeeze a patient by diuresing them and their NT-proBNP comes down and their GFR declines, then, you know, that may not represent actual disease progression in terms of worsening amyloid in the heart. Whereas on the other hand, if you have a situation where You diurese a patient and their GFR goes down, partly probably because of your intervention, but their NT-proBNP, for example, is increasing despite that. Then that really tells you that, you know, the condition is highly likely to be getting worse. And I'm just interested in your comments about the possibility of combining biomarkers, I suppose, in this sort of scenario.
So if I understand what you're saying is you're thinking of this as a surrogate for actually beneficial cardiac effects, not in isolation, but potentially with other variables. A part of that, I think the first step here is to say, what am I actually thinking I'm going to predict here, right? Like if I were to use this in a clinical trial, I'm, not going to look at the, the hard outcome because it's infeasible, blah, blah, blah. So I want to look at a surrogate, right? It's because it's going to be efficient. It's going to allow me to do this with, in a short time period, fewer patients. So do you make your argument for why you think you need to use a surrogate here, right? Because there's always some uncertainty associated with using a surrogate. But then I think the next fundamental question is, what is it meant to be a surrogate of? And if the idea is that this is a surrogate for cardiac outcomes, good. Charu will pull up here and she'll, obviously there's some concepts related to the validation of a surrogate that hold in any space. But I think the first thing you just need to be crystal clear on when we're having these discussions is what is it meant to be a surrogate of? What are you substituting this in instead of looking for the treatment effect on that outcome? And then that'll, I think, guide a lot of the next steps. And Charu's nodding her head yes, which is probably a good sign.
Thank you. Very, very helpful.
Okay. Now, I think we also have representation from several of the other companies that might have questions about sharing data or the process of this. Dr. Castano, Dr. Baylor, Dr. Vest, please do chime in here and comment on what you think might be the questions from your organizations or that you may have about what would be involved in the sharing.
I can chime in here. So I guess the first thing that comes to mind with ATTR specifically is given the rapid change in populations over time that we've seen, originating in the ATTR study to the ATTRibute-CM study and then Apollo B and now Helios B. And let's say, for sake of an example, combining all the placebo arms of those 4 trials to study some, renal question. How would we think about that as, it's a grouped population, but really at 4 different eras of time, right? And would we think about analyzing by era, by trial, by, as a subgroup analysis? How might we think about that?
I'm happy to speak to that. Julian may have thoughts as well. It's a great question, and I think it also speaks to the point that Dr. Alexander brought up about how because the standard of care is changing so quickly, like being nimble and able to learn from data in a timely way is so important. I think it gets down to the question we're trying to ask. I think if we're trying to understand an association between biomarkers and clinical outcomes, I think we'd want to know, is that a consistent association that holds up over different types of treatment? Or might it be that the association changes? I don't think we would know until we look, but we'd have to be sensitive to that possibility. I don't think we'd want to assume that things are the same over time, but I think we'd want to have the ability to interrogate that and see if these associations are changing or if they're getting at something biological that's more stable under different types of treatment.
And if I can ask a related question or make a comment, presumably having patients who are potentially at a different level of disease severity at the onset of these trials, partly down to the change in diagnostic patterns in ATTR amyloid cardiomyopathy. Presumably that to some extent is an advantage because you can see whether the associations hold up really through the range of severity of the disease. But I'm wondering whether that is the case.
I would think that would be helpful, but I'd love to hear James and FDA's point of view on that as well.
I'll cede my time to FDA. I'd love to hear you on that.
Got it. And I always feel funny. I think you're referring to me as FDA. I always have to be careful, right? Like these represent my opinions, may not represent anything other than that. Yeah, no, I think it's. I think it's generally true. The greater diversity, heterogeneity you have in a data set, and your ability to still see relationships, that tells you. I think that gives you more confidence. So yeah, no, I think it was a great question, and yeah, I think there's strength to having this diversity in terms of, yeah.
Can I ask just a follow-up then? I mean, that all makes sense, and I'm all for this type of approach. Just roadblocks I envision if I were to go back, to the leadership at BridgeBio, for example, and try to, pitch this. I guess, and this maybe is a question for Janssen, since you've already pioneered this. What were the main roadblocks that were encountered so that that we could potentially learn from that experience and use whatever tricks and tips you might have?
Well, for Janssen, we do have actually outside of this study, we have many other completed studies that we are part of what we call the YODA project. We actually provide a whole complete data sets to the third party who's going to be managing the data sets and for independent researchers who can access these data to do their own analysis. So for us, it's not a problem. The only problem, as I mentioned earlier, is nuance that because some of the analysis that we're asked to do is based on some endpoint that we have not yet formally analyzed for our study because it had not reached the sort of the event number or the timeline specified in our SAP. And so that was the only sort of roadblocks for us, is not able to provide at that time, but we did commit that we're going to provide all those analysis once, we had, fulfill our commitment to our, health authorities in terms of these endpoint analysis.
Certainly, this is at the concept something that is a proposal. But I certainly would be interested in the other data owners' questions that your organizations may have, or suggestions on how we could make this a stronger proposal. If there are proposals from your end, we'd certainly love to hear them.
I'll maybe just comment and a question. I mean, this is really is interesting, and the appeal of this is so clear. We've heard multiple suggestions today, as was highlighted in the presentation, of where this could be a really powerful tool to advance things for the patients. I guess what I would wonder about a little bit are the understanding how easy or complicated this is in terms of the resources. We can say this is a great scientific idea, and we can agree to the terms of the data sharing, but how easy or complicated is this and resource intensive to deploy the analytics? As I understand, you give a package that's then the stats group at the organization uses. Is this really straightforward, or is this something that becomes a substantial resource requirement?
From Janssen's standpoint, it's not very resource intensive because, we have the data specification, and because we have discussed it in advance, so we know exactly what data that would require to go into sort of, that package. And then it's just a matter of, setting up that specification and then run the data, run the code, because the code was given to us by, by James, right? So everybody, everybody sees that all.
I can share, and we actually put this in the IAS poster, but we did survey the participants around resource requirements, and I think it was less than 9 hours to take those steps that Dr. Tran referred to.
Is that is that 9 hours after the harmonization step, or is it?
That was mostly the harmonization step.
Oh, okay, great.
I'm going to have to hop in here and say we are at time, and I'm going to give Kristen just a couple of minutes to talk about next steps before I close the meeting.
Yep, thank you. So one piece I do want to highlight, while we've spent quite a bit of time this afternoon talking through how federated analytics could be an approach for this specific project proposal, we're very much at the point, as I mentioned, where we're coming up with these project ideas. We used this project as a case study for how federated analytics could be used, but it's something that we want to explore. Which of these projects that are of highest priorities, where is federated analytics the best approach for individual projects? And so this was a really nice case study because it's one that I think is ripe for that specific approach, but we are intending to evaluate across all of the different working groups, all of the proposals, which are the ones that are of highest prioritization to move forward immediately. And so this is the slide that I presented earlier today, but just as a reminder, we're hoping to, moving forward, make that evaluation in the January 2025 timeframe. So over the next 3 months, we'll be assessing the analytical strategy, feasibility for each specific project, with the goal that the working groups will then, across the board, be kind of rating how we move these forward. Part of that decision-making process before we get to really feasibility will be a post-meeting survey that you all receive tomorrow in your inboxes, really asking for feedback on the different project proposals. Again, it's a chance to share perspectives if you didn't in person, once you've had a chance to reflect and kind of think these through. This really does help us move forward and think about future initiatives as well. And then one last slide. I just want to, there we go, highlight that we are moving these meetings into an annual cadence, and so while it seems far off, we will be planning to reconvene these working groups formally in about a year's time, not on an election cycle, and so hope to see you all there next year. I'll turn it back to Isabel.
Excellent. So really an enormous thank you to everyone. I think today's meeting really shows that this is so much more than we ever imagined it could be when we first created the forum. And it really is due to everybody here and everybody virtually. We had 44 active participants who were panelists or speakers who really invested, as we've seen, an incredible amount of time and collaborative efforts to produce the kind of amazing work and discussions that we had today, and likewise a really engaged audience. I would just urge, as we see the opportunity with all these different markers that we want to consider, thinking about pulling those back to what has meaningful change and impact for patients, and bringing patients back into the center of the thoughts and discussions and engaging them. And I would really like to end by saying, I think James said it so well, collaboratively is the only way to achieve this. So I think we all have our work cut out for us, but it was so exciting to hear the potential and possibilities as we move forwards. And lastly, I would like to humiliate Kristen, our fearless leader. Without her, the forum would be nothing. And likewise, the rest of the ARC team, who behind the scenes have been herding, corralling, drafting, and making sure that this was a seamless event. So thank you all, and thank you all for coming.